Milton, sir. Sir, can you able to hear me, sir?
Hello.
Hello, sir.
Yeah.
Can you able to hear me, sir?
Yeah, I can.
Yes, sir. We'll start at 9, sir.
Yeah, we'll wait.
Yeah. Sir has already joined. Venkateswaran, sir.
Oh, great.
Milton sir, good morning, sir. Milton sir, good morning.
Sir, good morning. Welcome.
Yeah.
Ma'am, can we start? Purnima, ma'am.
Yes, ma'am.
Yeah.
Sir?
Arvind sir will join after 5 minutes. We'll start.
Okay. So good morning, everyone. I welcome you for this one-day pre-conference virtual workshop on image and video analysis, which is a part of 5th International Conference on Computational Intelligence in Data Science, organized by the Department of Computer Science, SSN. This pre-conference workshop has been organized by Machine Learning Research Group of SSN. This one-day conference virtual workshop has 3 sessions. First is Introduction to Medical Image Processing, and the second session is on Artificial Intelligence in Medical Imaging, and the third session is on hands-on with medical imaging using deep learning. I welcome Dr. R.S. Milton, sir, to give a welcome address.
Yeah. Thank you, madam. As was introduced, as a member of the Machine Learning Research Group of SSN, I welcome you all to this pre-conference workshop conducted as a part of IFIP approved 5th International Conference on Computational Intelligence in Data Science. I will briefly share a few words about SSN and SSN, Department of CSC and MLRG. SSN College of Engineering was founded by Dr. Shiv Nadar, chairman of HCL Technologies, as a philanthropic venture to give back to the society that nurtured him. Within a span of 24 years, SSN has grown into one of the top educational institutions in the country and has become autonomous in the year 2018. SSN offers some of the finest graduate, undergraduate, and research programs with accomplished faculty, world-class facilities, and a residential campus set on a sprawling 250 acres of sylvan surroundings. SSNCE is accredited by NAC with A+ grade. SSN Department of CSE is Anna University recognized nodal center for PhD program with 19 recognized supervisors. From SSN CSE Nodal Center, 23 have completed their PhD so far. 36 are currently pursuing their PhD. SSN Machine Learning Research Group was formed in 2014 with about 30 researchers. We conduct research in a broad spectrum of areas ranging from algorithms and theories of machine learning and deep learning to their applications in computer vision, image analysis, natural language processing, text analysis, speech processing, robotics, and analytics. SSN MLRG is carrying out projects of social relevance and has attracted funding from several agencies. MLRG has so far completed projects to the tune of ₹50 lakhs. MLRG collaborates with research groups across the globe. MLRG publishes their research work regularly in reputed conferences and journals, conducts workshops, conferences, FDPs, and invited lectures, organizes and participates in shared tasks with notable achievements, and also create corpus for general research. MLRG also envisions to be a leading NLP research group for Tamil and other Indian languages, coordinating the related efforts and disseminating resources. The High Performance Computing Lab of MLRG has the state-of-the-art research lab, providing an excellent deep learning ecosystem with high-end GPU servers and GPU workstations, network storage, and internet facility. And MLRG is organizing this track 1 workshop on image and video analysis. This is version 2. As was introduced, there will be 3 sessions. The introduction to medical image processing will be by Professor Venkateswaran, Department of ECE, SSN. And the second session is on AI in medical imaging. We are thankful to the 2 resource persons, Mr. Akash Narayana and Dr. Siva Teja Kakileti of Nirmay Technologies. And finally, we will definitely have a hands-on session on medical imaging using deep learning by Dr. P. Mrinalini of SSNCE. I once again welcome all the participants to this pre-conference workshop and wish you a very fruitful time together. Thank you very much.
Thank you, sir. It was a wonderful welcome address, briefing about our institution and the MLRG group. And let us start the first session. So the first session is Introduction to Medical Image Processing. This session will be handled by Dr. N. Venkateswaran. And there is a short description about him. And he has obtained his MTech degree in ECE from Pondicherry Engineering College from Pondicherry University and his PhD from Anna University, Chennai in Information and Communication Engineering for his thesis in the area of image compression. He is a fellow of IETE, senior member of IEEE, as well as a life member of ISTE, and he has actively participated in the different activities of IETE Chennai Center. He worked as a professor at Sri Venkateswara College of Engineering, Sri Perambatoor, and served over 13 years of experience. He is currently working as professor in ECE Department of SSN College of Engineering, and totally he has more than 23 years of experience in teaching, research, as well as in addition.
I think there is some network issue.
We are not able to listen to.
Yeah, there is some network issues, sir. So I'll continue, sir.
Yeah.
Dr. N. Venkateswaran obtained his MTech degree in ECE from Pondicherry Engineering College and and obtained his PhD from Anna University, Chennai. He worked as a professor at Sri Venkateshwara College of Engineering, Sri Perambudur, and served there for over 13 years. He is currently working as professor in ECE Department of SSN College of Engineering, Chennai. He has more than 23 years of experience in teaching, research, and in addition, 8 years of experience in industry. He is a BOS member for few other institutions. He has authored many research articles and book chapters, and he also co-authored over 100 papers in national and international conferences and journals. He has organized and conducted many FDP programs, international conferences, and national conferences too. He has extended his support in various academic technical committees and review panels. He has graduated 8 PhD students who are now at leadership positions in academia. Currently, 5 students are pursuing PhD. Also, he was supervised more than 30 master theses. His area of interest includes image and signal processing, machine learning, wireless communication, and optimization techniques. He also involved as a principal investigator in an ISRO SAC funded project. Welcome you, sir. I request you to take over the session.
A very good morning to everyone. Thank you, Dr. Mrinaldi, for a very nice introduction. Thank you. Let me start and share the screen. Is the slide visible?
Yes, sir.
Is the slide visible? Yeah, okay. Okay, let me start the presentation about this introduction to medical image processing. Why is this medical image processing so special? It's something that everybody will keen to understand, because this particular topic is actually patient specific and patient centric, meaning that people are under certain trouble. Any kind of a solution to this patient at this point of time will definitely help the patient so to recover from other pains and defects. So image processing is, you all know that it's a very nice signal processing tool that has lots of interest, including detection, estimation, analysis, and many more. Very deep understanding of information from externally obtained non-invasive way of taking images and then understanding from that. So image processing is a kind of a tool that every doctors and every doctors use, and this particular field of processing has come in a very big way. We all know that we take different modalities of images. So with the help of these different modalities, we can probably understand and detect certain irregularities that could be understandable with the patients. So again, these irregularities also is not same as with every other patient. So image processing is the effort of image processing has to be very important. So medicine is primarily kind of a tool that today we are all undergoing. So radiology is one of the physiological imaging tool modality, like modality like surgeons or the marines in this war of medicine. We make, as I told you, a lot of good decisions based on the available information, whether the information is sufficient or insufficient is something. So the success of any image processing is being measured by whether the patient is actually recovered very well, making use of these informations. Image processing actually gives, as I told you, identifies, measures, and also gives you some judgments on diseases, sometimes, it gives numbers to probably evaluate across patients. Of course, number of actions can be taken based on those results. Complete image understandings, of course, needs to be automated. Okay, so we need to have an automated decision making systems. So we need to have something called automatic diagnosis, where computers can perform number of functionalities for diagnosis, of course, and with robotic system into process of surgery and other things. So we need to have a good control over the CAD tool that might get developed through this processed images. So one of the very important processing step that is generally followed in medical image processing is what is called a segmentation and registration. So image segmentation, you all know, like you have to segment region of interest and then assign labels for each of those region of interest. So basically, images consist of pixels, and labeling pixels for region of interest is something done through certain algorithms. Today, extensively, our machine learning group of people employ machine learning algorithms to label each of those pixels that are present, whether the pixel of interest is diseased pixel or it's a normal pixel and things like that. So number of segmentation approaches are always there in image processing. You all very well aware. The very basic approach is to segment based on certain threshold. Just I've shown you here, there is a gray matter and white matter. So we basically convert into a binary kind of an image based on certain threshold. And other segmentation based approaches are based on, for example, edge based segmentation, and then sometimes region based segmentation. Similar pixels can be grouped, and we can use some kind of a clustering techniques, unsupervised kind of machine learning technique for segmentation. So these are different models. I'm just bringing out different models for different algorithms for doing segmentation. Of course, we cannot leave out the importance of artificial neural networks in the process of segmentation. So we can design number of algorithms, techniques for doing this segmentation approaches. Of course, segmentation can be with reference to thresholding technique. You can have a global threshold and what kind of a threshold which you can use. And the basic image processing techniques will tell you that there is something called Otsu threshold, an optimized value of Otsu thresholding technique and things like that. So those who are slightly familiar with basics of image processing, you will understand why segmentation is highly important in this process of image segmentation. The next, hello. Yeah, the next important process of image processing is something called, yeah, we can put off the audio further, Mani. Okay,
Sir,
yeah.
I'll take it, sir.
Yeah, thank you. So the next process of image processing technique that could be used is what is called this registration and visualization of images. So medical images have to be visualized. So we have to keep the picture in mind so that we could be able to see the progress of the spatial, whether it is in time domain, we see the progress of disease, or spatial distribution of the disease, and things like that can be seen if you could go for visualization. So visualization is important as well. In order to do this visualization, we need to do this kind of what is called registration. Registration is basically to combine images from or trying to keep one-to-one relationship or mapping between one modality of data with the other modality of data. So you all know that registration has to establish the correspondence between the image pieces across a set of images, and then you may have to use some other transformation model to infer any kind of correspondence between these images. So we need to register images, because computer aided and computer aided detection and diagnosis using machine learning algorithms can definitely help physicians to interpret medical imaging findings and reduce, of course, interpretation times, as well as mistakes that could probably happen. So this idea of this image processing work is to help doctors, right? So in order to get a very good evaluation of the patient through number of images that they will be able to obtain from the patient, through non-invasive way of understanding. So now doctors look at many types of image modalities. So medical imaging has number of modalities. So based on the source of energy used imaging, external or internal, or combination of the hybrid kind of this thing. You have X-ray radiographs, including you can have plain X-rays and X-ray scans, fluoroscopy. Some of the X-rays today make use of fluoroscopy. So X-ray, CT, ultrasound imaging, and this optical transmission, as well as based on acoustic signal. There are a lot of imaging is taking place today. That's the last part of this external imaging. Of course, you're all aware of this nuclear based medicine. So nuclear medicine, you introduce certain radio tracer and scan the portion of the body, so that you get something like this PET image, nuclear medicine image, positron emission tomography images, as well as the SPCET, single photon emission tomography images. So some of these images reveal very good information of the patient, body of interest. So of course, combination of these two gives you this fMRI, PMRI, optical fluorescence imaging, and as I told you, the optoacoustic imaging is also coming in a big way. So a vital contribution to the overall diagnosis of the patient and help the decision. All these modalities are helpful for the overall decision of the treatment of the patient. So I've just shown you a number of images. So mammographic mass classification. So all this list of images that I've listed from top left to bottom right. They all today we all use deep learning as well as machine learning models to process them and then make decisions based on this. So mammographic mass classification, segmentation of lesions in the brain, leak detection. Then you can have retinopathy, diabetic retinopathy classification, prostate segmentation, nodule classification, and number of such areas of application is already there. So the question is, why do you need machine learning algorithms? Machine learning algorithms are required. Deep learning algorithms are required to
participants are requested, sir. Just a minute, sir. Participants are requested to unmute yourself. Lakshmanan, kindly unmute yourself. Mute yourself.
Yeah, I think you can have a control, madam. You can mute.
No, sir. No, sir. It is with the conference people.
That's
not
okay.
available.
That's okay. Anyway,
I'll make that one, sir.
Yeah, no problem. So this machine learning algorithms give computers the ability to learn without programming explicitly, and of course make reliable predictions on the image data that is given. So one can through machine learning algorithms, one can optimize the performance using the example data as well as the past experience that we have. So in this, we use number of statistical measures. The computer science people can efficiently design algorithms to solve these optimization problems. There are different types of learning methodologies, what is called as the supervised learning, unsupervised learning, and reinforced learning. In the course of our discussion, we'll see some of those definitions. And learning is needed when human expertise does not exist. For example, this is typically for other type of images, but particularly for medical field, we need to have doctors who can interpret images very efficiently. So getting a doctor at a time is very difficult. So we need to have to replace the human expertise. Of course, I told you earlier, the patient to patient kind of variation that will probably available, that needs also has to be learned through the machine learning models, and solution needs to be adapted to different cases. We need customized solutions, okay, things like that. So learning is not needed when you actually want to have all this features absent. So something, there's a fixed kind of data, we don't need to do. So in a typical medical data that varies from patient to patient, we need to have all this kind of learning. So as I told you, there is major class of learning is supervised, and the basic work that is done through the supervised learning is to do classification. So in a typical classification, you will have set of training sets and what are called classified labels. So you will have what is called labeled data. So this kind of label, for example, typical medical data might contain lesions, for example, and areas that are to be marked as diseased areas, and you can interpret diseases if you have region marked as disease or undiseased region, and you can have a training set for all images. So this is a classification task generally given to supervised learning. In a typical unsupervised setting, the algorithm is generally of what is called clustering in type, wherein the training data is not generally labeled. So in the previous case, you need labeled data. In this case, you don't need to have labeled data. The output label is basically generated using the training during the training. So similarity across features of training examples creates basically different classes. Now, simple binary class can be yes or no type of class, and then you can have multiple classes through. If the image contains different types of diseases, and then you can come out with classes, multiple classes for the same image. In a typical measurement or identification or evaluation of tumor, the input features could be a size of the tumor and rate of growth, and some of them can be used as a features for training the machine learning algorithm. So if you don't have proper labels for this, so if we assume all the labels are tumor, cancerous in nature, then you can come out with different class of output. So you, based on size of the tumor and rate of growth, so there are multiple classes you can perform and see the growth of this tumor over a period of time, and maybe you can look at how this process, if you have multiple patients, for example, you can come out with the treatment process, whether it is effective or not, and things like that can be understood. Okay, the most simplest kind of unsupervised learning is what is called k-means clustering algorithm. So this k-means clustering, if you extract features that we have just told you for tumor types, you'll be able to cluster into different classes. For example, the cases could be this state of tumor growth beginning, or level 1, level 2, level 3, multiple levels. So you can get these grouped into different levels, and if a particular patient belonging to a particular label based on the intensity or the extent of tumor, then it will get associated to the particular class out of the 4 or 5 classes that you wanted to cluster them. So now, typically in a classification stage, any new class that is actually a new data sample that you wanted to present to this clustering process will find what is called distance with the existing centers of these classes. So we calculate the distances. I mean, there are multiple types of distances, you all know. Very simplest type of distance calculations, what is called as the Euclidean distance. So any data that produces minimal distance from a particular center will belong to that particular class. So the advantage of this k-means is it is very simple, and it can also work with large data size, right? And large. But however, the a priori number of classes that is available in a particular image data set may not be available at the beginning. So again, this algorithm depends on the choice of the initial centers. So again, very complicated data with some overlapping features cannot be identified by this k-means algorithm. So we get into some other types of algorithms are known as the coherent self-organizing maps. So it's basically a high-dimensional complex data clustering tool, which basically makes use of this what is called the neural network learning. So the other method that is seen in the literature is what is called as the ISO data, an iterative method that uses Euclidean distance as a similarity measure. I mean, this is it is an iterative method. So you all know that k-means is also an iterative method. So makes use of this Euclidean distance similarity measure into cluster into different classes. So simplest type of again, another classification algorithm is what is called k-nearest neighbor algorithm. So in this case, what we understand is that regions with similar characteristics or features will be available in a group. So we have to understand the neighbors, and based on neighbors class, the particular neighbor pixel class, the current pixel will also be classified. So again, this involves distance calculation, and any class with the minimum total distance will be associated with the new pattern. So the main advantage of k-nearest neighborhood is just like neural networks, does not require knowledge of probability distribution. Is also less complex, computationally less intensive. More computationally intensive classifiers exist that do require exact knowledge. So we look at more complex patterns which mimics the function of the brain. So this is how brain basically learn, the neurons learn. We can mimic that operation through this kind of computational model. The computation model is basically theoretically well studied in what is called artificial neural networks. What I'm shown here is the simple neuron, which consists of this input from other neurons through, the neuron senses these inputs through what are called dendrites, and combines all these inputs through a cell body, sends it down to a next neuron through what is called axon. So you all know that brain consists of millions of neurons interconnected, and what is shown here is a simple neuron. You can think of interconnection of multiple neurons in producing or learning and storing patterns within the brain. So this is essentially what is being mimicked through a simple computational model that I've shown you here. For the cell body representation is shown in red color, where there is a summation of all the inputs weighted by certain weight is basically used to used to, it's subjected to some kind of an activation function, and then the output is released. And this particular output is basically the kind of output that we wanted to obtain through training. So there's a nice book on this neural networks written by Fawcett, Fundamentals of Neural Network Architecture and Algorithms. Okay, so this can be used for any course. So of course, you can have complexity of the neural network comes into picture. You have a single layer network which decides based on the n input features x1 to x1. You can have multiple classes, y1 to ym. So these are single layer networks. You have a multi-layer networks. There are lots of hidden layers. Now these hidden layers store weights during learning, and there are a number of algorithms to adjust the input values with the output values during learning. So we have number of learning algorithms. You all know that very famous algorithm is what is called as the backpropagation algorithm. So the architecture that I've shown you, either a single layer or a kind of a multi-layer network. So so it sometimes it's shown that three-layer network with some kind of a input and hidden layer, output layer network is sufficient to probably simply you'll be able to make a simple neural network. But a rule of thumb suggests that the number of hidden layers must be less than 1/10 of the number of training examples. So more the number of layers, hidden layers, of course, there will be other complexities associated with the classification and detection. So so we need to restrict and go by the rule of thumb to probably choose the number of hidden layers. So majority algorithms designed are, because I told you, or backpropagation or gradient based algorithms. You all know this gradient is can be computed for the kind of the loss function that you will probably choose for learning this algorithm, and gradients can be computed. These gradients, if you can pass through what are called as the negative value of the gradient, then you will reach to what is called as the very optimal value of the weights. So there will be minimal errors in representation. So as I told you, if you don't choose the parameters of neural network, the network can be said to be overtrained with the training data, and sometimes it may do very good with the training data, but however perform poorly for any new set of data that is applied to the neural network. So in a typical neural network model, we can put a number of input parameters like the symptoms and any biochemical analysis features, or some of the features like whether the patient's age and gender, whether he's smoking or not. All these things could be a kind of an input to the learning model, and you can probably come out with a decision whether the person has has a problem or not, whether it is the disease is positive or negative, or it's uncertain and things like that. So so neural networks are an excellent choice. So again, artificial neural networks, of course, I told you, can store patterns like brain stores information that you see since childhood, and maybe you are able to associate each object with a class. So by probably learning through this pattern recognition model, as you all find that the charts in front of you might come out with different shapes and different heights, maybe colors, but your brain will be able to find out and see whether it can be classified as a chart or so. So as I told you, the unsupervised model coherent self-organizing map. So this K-maps, coherent maps, are heavily used in biology and bioinformatics area, where clustering of gene expression data is extensively used. Okay. So this is the architecture of a coherent self-organizing map. It inputs neurons map in n-dimensional pattern, and there is a output is a competitive. There is a winning neuron coming out of the so many output neurons. So output neurons with similar patterns are arranged to be neighbors. Some output neurons might store more than one pattern. So this is okay. So object classification arranged in one-dimensional data. So passing data kind of a one-dimensional. See, coherent is basically a 2D kind of an array setup. The disadvantage of 1D array is that some objects may fail to get recognized if we have similar features which are not far from each other. So that's why coherent self-organizing maps uses what are called as for example, 2D information. So these are different classes with which these inputs are classified. I told you an example of a chair. So the other important machine learning algorithm used for classification of input data is what is called as a support vector machine. The idea here is that you need to design a classifier that the classifier has largest margin between two classes. So we come out with what is called as an optimal classifier, which has a maximum separation between two classes of data. So we come out with these equations of those classifiers. For example, WX transpose plus B, 0 is the line of interest, and the other two lines which belongs to class 1 and -1 other class. So they come out with another line. So I mean, if you can able to maximize the separation between these two lines with the center line, then you are actually doing a very good classifier. So there are various ways that this line can be drawn, but however, the one which has the maximum margin can be said to be the result of these SVM. So one can train SVM classifiers with these two class of data. So essentially, you will have a linear separation. The one that you saw now is a linear separation. So these are the input to the model. You will have data and its corresponding class. Now here we are using a binary class with the two outputs only, either -1 +1. See, the SVM is to find the coefficients of the line and the intercept W and B of the hyperplane. For example, if it's a more than two dimensions, you get into a plane. So you have to find WX transpose to be equal to 0. So this is the requirement. So optimization problem can be solved and can be formulated through a decision function, which is written here. So it is sine of alpha I, summation over all classes, alpha I, yi, the data, and transpose data plus B. So this can also be used for multi-class classification. So in a typical multi-class classification, we can initially start with classifying one label with the rest of the other labels. So it's called one versus all classification kind of setup. You can extend this with each of those labels, and then you can come out with multi-class classification. So the problem exists in SVM is that if the data which is available is not linearly separable, then we look at what are called certain kernel tricks. So these kernel tricks, we use some kind of a kernel like radial basis function kernel or a Gaussian kernel to translate the data from one feature to one domain to another domain. So this is called as the kernel mapping. So, or a kernel trick. So kernel exactly maps the input data space into a different data space, wherein in the modified space, the data can be linearly separable. So this is the kernel function phi. So this is the requirement of a kernel function. So the kernel function should be separable, and it should be phi xi and phi xj. So if you see on the left hand side, the data is quite nonlinear. You need to have have to convert them into a linear data by using this nonlinear mapping function phi. So now what we did is we tried to use some kind of a we did a study on use of SVM for example, studying psoriasis skin disease identification in the using SVM model. So you all know skin diseases tend to be itchy over the body. So psoriasis exists on a common chronic inflammatory disease of the skin caused by scaly patches. So you can have a very nice image of this skin. Of course, it will come out with different colors and things like that. You can extract very easy features from the images, and we can subject it to what are called SVM segmentation. So this is essentially we use something called feature scaling in doing this. So I will just get into the methodology here. So the input image is taken. We do to remove noise. We use some kind of a median filtering, and we use what is called Markov random field algorithm for segmentation. So after segmentation, we extract features and give it to what is called SVM classification. So through this image that is segmented, classified, you'll be able to know the disease type as well as the growth rate of this. So we have taken this images from this data source. The process starts with the collection of database images. Of course, I said median filtering, segmentation using MRF, classification using SVM, and we do this bifurcation. Each segmented image is bifurcated into 9 parts. This is a process we used for easier segmentation of the disease region. So again, we identify disease portion, and then once disease portion are identified, the temporal growth of this region will also be understandable. So these are some images. So after segmentation, you get the figure number 3, and then figure number 4 is your segmented binary image, and then you get what is called SVM segmented RGB image. So this we try to do as with the help of students with the segmentation and bifurcation process, and then the image relatively obtained, and the region of growth can be understood from region of growth can be understood from multiple temporal images that are supplied to this. Now, this is the different types of psoriasis images. Just showing you there's certain images. So these types of images, what type of psoriasis is also can be identifiable through this SVM network, SVM. Okay, so we had the 2 different diseases that we have in the psoriasis. We had evaluated certain features and then subjected to for different patients. For example, I1 to I16, and then the disease identification is finally carried out. Now, in the second example, we used what are called detection of diabetic retinopathy using certain machine learning algorithms. So here also we considered what is called as a fundus image, color fundus image. So we extracted what are called GLCM and GLRM features, and of course the loss features. So after doing some simple pre-processing, we used what are called text to feature extraction methodologies, and we subjected to classification through either SVM as well as what are called the K nearest neighborhood algorithm just now I have outlined. So we extracted number of features, GLCM features, starting from energy, entropy, homogeneity, and different moment values and standard deviation values. And we have listed out some of the differences between the normal and abnormal. For example, the contrast value is quite double for a normal with respect to abnormal. So some of these features have a very distinguishing characteristics. So we exploited this nature of the data for classification. So Here again, other features, correlation-based features, of course, including prominence, inertia, dissimilarity, difference variance, sum variance. All these features are some of the regular features that are generally available in a GLCM feature extraction methodology. One can follow this. This, of course, tells you the variance of each of those features. This is called as a box plot. You can have the box plot definitely gives you a number of informations related to the maximum value of the observed, the minimum value observed, and for example, the median value, the upper quartile, and as well as the lower quartile values. So we also extracted what are called GLRLM features. And also we extracted what is called loss features. Loss investigated the 3 text feature generation methods. They are called co-occurrence, correlation, spatial statistical features. So these values are also used for finding or subjecting this into a multi-label, multi-feature, for example, dimensional features which are generated out of all these features, and then we subject this to, for example, classification through SVM, KNN. So along with this, we also took the 2D radon transform of the image. So we again got number of higher order statistical features. Now this is called the cumulant features, right? Cumulants and sinogram images. So again, we have listed out features for abnormal, normal categories, and then also we did it with the K nearest neighbor classifier. We use the distance metric as city block distance, and in this case, and then we come out with accuracy values, both in terms of K nearest neighbor as well as SVM classifier. So we understand that SVM classifier is a supervised classifier based on statistical learning theory. So we used, of course, the kernel functions I told you, because the data was quite nonlinear. We need to have transfer this through a radial basis function into a different, for example, space, and then we did classify that. So some of these classifier accuracy is also given to you for understanding, right? Now in machine learning approach, the other very important technique that is used, what is called as the decision tree algorithm. Okay, so these decision trees and regression trees hierarchically create some structure that is used to analyze as well as classify data. This is also very good in medical imaging field also. So I'm sure you all know this decision tree algorithm, so I will skip this. Of course, a number of measures are used for finding the, for example, finding the hello. Okay, so we use kind of measure called entropy. High entropy means the data is more random. Less entropy means the data is very smooth, as well as it has got less, what are called impurities. That is, so you'll be able to analyze data based on this entropy measure. Through this entropy measure, we can get something called as the information gain. So in a typical decision tree, you have a parent and there are child nodes. So you can measure the entropy of the parent node, measure the entropy of the child node. You'll be able to get what is called as the information gain over this network. So there are many other types of decision tree algorithms, which are ID3 and CART. CART refers to classification regression tree, and C45. Some of these techniques are available in a typical machine learning books. Now, decision regression trees can overfit the data. So overfitting again is not good. So we need to prune these trees. So trees are first expanded to large depth and then pruned and collapsed according to the quality measure that is. Okay, so another technique that is generally used in machine learning algorithms is called as the random forest classifiers. Okay. Using subset of data and subsets of input variables, we can form the tree. Performance of the trees are tested using the data not seen by those trees earlier. Finally, the output of the random forest tree to a new input will be the average of the output of all trees that you feed as an input. Now, a random forest is supposed to give good accuracy without serious overfitting. So some of these things can in general be used for machine learning algorithms. How to handle the data set is something that we predominantly look at. So data set can be generally classified into what is called as the training data set as well as the test data set. So test data set. So in a typical training data set, you have to classify either training and testing, and in another approach you can have reserve some data for validation and tuning up the model. Some of you people who some of the researchers already will be using these techniques. I'm only just revising them for benefit of how this can be done. So people can also use in order to effectively use the available data. If the available data size is not sufficient, you can go for some called technique called cross validation, in which case you can regroup the data into multiple segments, and then you can have, for example, n-fold cross validation. So the data that was used for initially for testing now again put back into the process, and you generate a new data for testing. So so this cross validation is generally used in case that we don't have enough data for your, or if there is an overfitting and things like that happens. So based on the available results, available results can be measured based on a confusion matrix. Now, confusion matrix can be for a two-class problem. You can see the actual class versus the predicted class. So, if the predicted class is something and an actual class is something, then you are actually, if they are different, you are getting into false locations. So, the predictions could be same as the actual class, then it can be called as true. So either the disease is present in both the types, both the prediction as well as the actual, then it is true positive. If the prediction is also negative and the class label is also negative, then it's called as true negative. Otherwise, it's called as the true positive. So this particular confusion matrix will give you the quick assessment of whether the algorithm has given you good results or not. So based on this confusion matrix, there are a number of other measures can be computed. One is called the accuracy, sensitivity, specificity. Some of these formulas are you can generally understand from the literature. So I'm just, for completion's sake, I've just put it here. Again, in order to check the validity of your accuracy, and we go in for finding what are called receiver operating characteristics, where the false positive rate and true positive rates are plotted in X and Y axis respectively. The area under the curve basically tells you the accuracy of this curve. A good ROC is the one that rises early so that high sensitivity and specificity are achieved at the same time. So basically, this kind of ROC curves are used for comparison across many algorithms. Okay, so again, if you have large number of features like the one that we used for the fundus image classification in retinopathy, we had a number of features, and we have to subject all these features into what is called dimensionality reduction. So we need to probably obtain features that are more distinctive in nature than the whole lot of features. So we use some kind of a principal component algorithm for extracting the best features out of the given data. So as a matter of fact, we have actually discussed a number of machine learning algorithms that can be used for along with the medical data for classification and recognition of diseases. I mean, I would like to again stress the use of multi-layer neural networks along with support automation and regression trees are one of the major techniques that are generally used for this kind of classification. So again, we talked about this problem associated with machine learning. Machine learning algorithms are susceptible for overfitting. So in such case, we can opt for some kind of a n-fold cross-validation. So there is, of course, the importance to reduce the dimensionality of the data and things like that. So since I've just touched upon some of the major machine learning techniques, let me also quickly go through the deep learning models currently being employed for medical image classification recognition. Those of you who are already familiar with the deep learning models, there are a number of deep learning models that are already available in the literature. Either you can use convolutional neural networks or recurrent neural networks in the form of LSTMs, long short-term memory networks. There are generative adversarial networks and convolutional autoencoders. And more recently, we have something called graph convolution networks. I'm not going to the details of all the networks. I may probably touch upon one or two of these networks. All these networks can be used for a number of applications, including lesion segmentation, lesion detection, learning methodologies used here are supervised or semi-supervised. Of course, number of other disease detection methodologies can be used. Okay, so is it okay now? Is it visible?
Yes, sir. Yes,
sir.
Visible.
Yes, sir.
Okay. So the convolutional networks are a type of artificial network which is widely used for basically recognition and segmentation. It has a lot of convolution and pooling layers, which are actually reduces the size of the input image into number of feature extracting layers, and then finally you will have what is called as a fully connected layer, and then this learning algorithm goes from forward to backward multiple times, and then the image features are learned across this network. So it can automatically detect important features without any human supervision. This is the one advantage. Earlier we have to extract features and feed to the network. Now it can be said that so it takes advantage of what are called local spatial coherence of the input, which allows the process to extract fewer number of weights. So this is essentially used generally developed for not only for medical images. It is used for initially for a number of images that are object recognition, object detection, and things like that. So these what are called the models that are already developed can simply be used for our purposes. The other network is generally used is what is called GAN networks. That are called as GAN networks are generally called as generative adversarial networks. This kind of networks learn to generate from a kind of a training distribution with a particular distribution with a set of real images to two entities through these two entities, which are called as a generator network as well as the discriminant network. So these two network adversaries are constantly learn throughout the training process and try to produce these outputs, whether it's a real image or a fake image and things like that. So this is the model for a convolutional graph convolutional network. So traditional representation of image is what is shown here, 2D representation. But as you go up, you can look at the medical images, for example, fMRI image of the brain or EEG data. can be can be modeled as a network. So for example, for example, this part A of the network that is shown at the bottom of the layer, basically used for understanding the brain response, and then the other network B basically used for analysis of emotions. And these graph networks have graph neural networks are used for learning many of those features in the image set. So what we did is we tried to understand the evaluation of sleep apnea is kind of a inadequacy in this. As people require good amount of sleep, and sleep apnea is a kind of a disease that generally used present with all of us. Sometimes individual does not consciously wake up, but the brain has to start breathing again, which disturbs the sleep quality. So this OSA, obstructive sleep apnea, leads to deterioration of health. So if we don't get a proper sleep habit, so this is to detect this sleep apnea through other methods like polysomnography analysis being used for understanding whether a person is snoring, etc. So this is what we did is we took some EEG signal and then we analyzed through the sleep apnea behavior of person. So the idea is to develop the prediction model for analyzing normal and abnormal sleep behavior of a person with respect to and compare with the clinical ECG data. The implement automatic diagnosis methodology for obstructive sleep apnea with the ECG data, determine the optimal model for this sleep apnea detection. Of course, evaluate the performance. So the students used a data set consisting of 70 records. It's a digitized version of ECG. And then ECG signals are digitized at a frequency of 100 Hertz that varies between time. Okay.
Total industry, we give them leave, their total industry work for 1 year, 2 years and then come back. That scheme is also there.
Okay. So time series sequence data will be generated for each ECG beat, amplitude, co-currents stored after detection. So we used based on this QRS complex evaluation of the ECG data. So we took this ECG, we did some this QRS detection, RR interval detection, and probably used deep learning based classifier. We used LSTM, GRU, and CNN. So we could able to differentiate or detect into two classes whether there is a normal or a sleep apnea detected. So we used LSTM, we used CNN for this process. I've shown you the models for each of them. So this was developed using Python and neural network libraries Keras. We used data into 70% for training and 30% for validation. So we present to you the model summary for each of them. CNN model summary. As someone who already aware of these models. So LSTM is used, for example, 150 epochs, both CNN also. Number of fully connected layers is 1,2. The dropout was fixed as 0.5 for LSTM and CNN. We used different activation functions and things like that. So the result of this I've just shown here. The accuracy, what is there in the first column of this tells you that the LSTM network has a better accuracy. So with an F1 score of 0.81 and things like that. So this was a study by again, we also did three-class classification of retinal disorders using CNN. So this is the what are called as the the OCT image, optical coherence tomography images of the eye. So what is shown is the normal eyes OCT, and there is DMD type and DME type. Okay, there are different disease. Diabetic macular degeneration, and the other one is DME. So we have these features, and we were able to classify into, for example, this is normal as well as into, that is why I've shown you these two models. So some of these diseases have differentiating behavior while you look at these layers of this coherent tomography image, OCT image, deposits in between RP and Bush membrane. There are a lot of layers in this particular OCT image. So outer nuclear layer thickness actually decreases. There is a photoreceptor degeneration is there. There is a serious retinal detachment. So understanding these features, we allowed the CNN to probably learn these features by itself. So we use some 12,000 images, 4,000 in each class. We also did some kind of augmentation by rotation as well as translation. Then we had the process of noise removal after denoising. Okay, so drusen macular degeneration, diabetic macular edema are the two DMD and DME. And normal eye, we after going through this filtering operation, we get a better signal in terms of PSNR. We evaluated through different denoising filters, and we finally said that something called as a Kuan filter will do. Then we constructed CNN architecture for the denoised image. The input is 227 by 227 denoised image, and it's subjected to all this pooling and convolution layers. So across this, and finally the number of classes here was decided based on the learning rate. So these are different layer sizes and kernel sizes that were used. Okay, so number of trainable parameters is, though it's large, we were able to train with the GPU system available with us. So these are the accuracy values which are shown here, the training and validation accuracy. These are different layers. You see the visualization of the layer. I think I can zoom out and see the features extracted. Each layers can be seen very easily. So we also did a lot of optimization with respect to CNN, hyperparameter optimization, hyperparameter tuning methods are used. Okay, so and then the results of this based on this tuning, we go in for choice of this optimal parameters with respect to activation function. We used ReLU. We used the gradient descent optimizer. The learning rate was finally fixed as 0.01. Number of epochs were finally decided as 100, and a batch size of 64 was used. This essentially gives you the accuracy and training loss patterns, plots of this algorithm. So we also did this 3-fold or 5-fold, 7-fold, 10-fold cross validation. So as the cross validation increases, the the accuracy also did not improve. So we probably stop at, for example, 3-fold cross validation itself. So these are the confusion matrix because it's 3x3 label. So 3x3 confusion matrix. So we had number of confusion matrix for actual disease. This is a label, predicted label on the first column. On the first second row, you have here the actual values, right? So we get into this confusion matrix based on this and formulas that was given earlier. We computed the sensitivity and specificity values, and the accuracy stands finally with the denoised image with 97, something like 97.47%. So we also set up the other networks in the literature, LXNet and LeNet. So these are fixed networks, so we do not modify. So the proposed convolution network for this is given you a better accuracy and bringing out, for example, the better accuracy in terms of. So I think I think I have only all this. So if there are any questions, I'm ready to answer. I think we'll spend about next 5 to 10 minutes to discuss any of the questions that the audience might have.
Participants, any doubts or questions?
This is being a machine learning group, and I need to probably spend a little time on the algorithms that relate to machine learning and deep learning. So some of the examples also under this each category was shown. Maybe is there any questions on all this?
Sir, good morning, sir.
Yeah.
Sir, to obtain the medical images,
Hmm.
whether we are in need of any ethical clearance?
Yeah, generally, anybody who works in medical image, because if it's patient specific and things are patient dependent, the patient may not willing to disclose the identity of the patient. So most of the time, we need to have a clearance from the ethical board. There's a ethical clearance committee will be generally available in any institution. So comprising of including doctors. So you have to take ethical clearance, but some of the data sets that are already available on the network, for example, something that I've shown you, does not require any ethical clearance because it's already available as a database for doing our experimentation. So ethical clearance is not required in such cases. So, but however, if you go to a hospital and you are trying to do a new analysis, extracting a signal from patients, then you need to have ethical clearance. Hope I answered your question.
Yes, sir. Thank you.
The idea of ethical clearance is that there is a committee that actually looks into the aim of the experiment and whether it is ethically correct or not, whether you use the patient data correctly and things like that. So any other questions?
Sir, this is Mrinalini.
Yes, madam.
Yeah, you have applied for the retinopathy features. You have applied some PCA, principal component analysis.
Yes, madam.
So the purpose is to reduce the number of features, no?
Yeah, generally PCA is a dimensionality reduction technique.
Yes. Whenever it is a deep learning model, the model says when the number of features is more, the model will be more perfect. Why we want to reduce the number of features in the case of machine learning algorithms?
No. See, the size of the features, dimensionality of features is actually higher the dimensionality for a machine learning algorithm. You are talking about what is called deep learning algorithm.
Yes.
In a machine learning algorithm, it is a multi-dimensional space. In a typical multi-dimensional space, if you increase the number of dimensions, then the data will sit at odd places. Okay, so the grouping, either classification will become difficult. In a ML setup, I'm talking about an ML setup.
Okay, sir.
Okay, so it will lead again to what is called this wrong detection, and okay, so that is why.
Okay, this reduction will reduce the overfitting or something like that.
Exactly. It will reduce the overfitting.
Okay. Then instead of going for a large number of features, can't we, because in machine learning, we people are deciding what are the features to be taken into consideration.
We conduct something called
that can be done, no?
Yeah, there is a way of computing what is called significant features using something called significant test. In statistics, we have a student t distribution, kind of a significant test computation is there. we can find out what is the significance of each of those features, and then make use of the very few features that are supposed to be more significant.
Okay. Okay, sir. Thank you.
Yeah.
Any other questions from participant side?
Any more questions? So again, with respect to this test of significance or significant test, we need to find out the features that are supposed to yield best result. So in terms of accuracy and other things. So we need to conduct this test of significance.
Okay, sir.
Any other questions or queries? Those who already work with medical images or any image. Hope everyone is very clear with what you said, sir.
I don't know, madam, this 1 hour, this thing cannot clear everything of everybody, but it's only an kind of an introduction, and make somebody who's not already into machine learning. I know many of
yes,
the
but
group
it was a refreshing session
is
for
kind of
those who work with.
Yeah, this is I mean, we cover each technique in each class, so we have
Yes,
to cover
sir. Yeah.
Okay.
Okay, thank you so much, sir.
Yeah, thank
Actually,
you
this
very much.
session gave us a very brief introduction to medical imaging, as well as it was an overview for all feasible learning methods to process those medical images or any images with relevant all medical samples, what you have been studied so far. And definitely it would be a refreshing and insightful session for all the participants, those who already work with images. Thank you so much, sir. Participants, please could you please fill your feedback for this session. The link has been posted in the chat box.
Can I unshare my?
Sir.
Yeah, I
yes,
can.
Sir, your presentation.
What do you want?
So will you sharing your presentation?
Yeah, no, I'll send it to you. A PDF version, I'll send it to you.
Yes, sir.
Yes, sir.
Yes, sir.
Okay, no issues.
Thank you for your time, sir.
Thank you, ma'am.
Thank you so much, sir.
Thank you. Can I leave?
Yes, sir.
Yes,
Sure,
sir.
sir. Thank you.
Okay, thank you.
So participants, please fill the feedback form for this is session 1, and you can join this next session at 10:45.
You can join with the same link. And this feedback form will be taken for your attendance, and certificates will be produced based on the attendance. So I request everyone to give your feedback. The feedback link is available in the chat box.
The next session will start at sharp at 10:45 on the topic artificial intelligence in medical imaging.
Hello, PM. Arnalni. Hello, good morning, Dr. Shiva.
Hi, Dr.
Yeah, good morning.
Good morning.
Yeah, thank you for accepting. This is Chitra. So I was reaching out to you. So
Hi.
yeah, thanks for accepting the invitation.
Akash also joined, and yes.
Hello, Dr. Chitra.
Yeah.
Akash.
Hello, Akash. Yeah. Thank you,
Yeah.
both of you, for accepting our invitation and joining here. Yeah. Mrinalini?
Yes, sir. Yes, ma'am.
Yes. Dr. Mrinalini is the organizer, so she will take over. Yeah.
Thanks.
Good morning, sir.
Good morning.
Thank you for accepting our invitation, sir.
It's our pleasure.
Shall we start at 10:35, sir?
Sure.
Now only the last session got completed. Told the session starts by 10:45. Can we wait or we will start, sir?
Sure, we can wait for another 5 minutes or so.
Yes, sir.
Right.
Thank you, sir.
Hello, Shiva. Hello.
Yes, Dr. Chitra.
Yeah. I thought while we are waiting for the audience to come, we can. I just thought I'll discuss regarding is Niramai having any tie-ups with academic institutions, or do you hire from the academic institutes? I just wanted to check with you.
Yes, we do hire a lot of interns every year. I think every semester also we do hire.
Yeah.
So, and we are in collaborations. I mean, we have hired interns from BMS College from Bangalore. In past, we have hired interns from IISc, IITs.
Okay.
Different MIT, Manipal Institute of Technology.
Okay.
These institutes we typically hire.
Okay.
So definitely, if anyone is interested in internship, they can write to my email address or Akash email address, which is in the last slide of our pitch.
Sure. Okay. Yeah. In fact, I was trying to get Geeta for at some point in time, but because as a as an entrepreneur, so it would be interesting to hear the entire journey. You are also one of the co-founders, right?
Yes.
Of Niramay. Yeah. So,
right.
Yeah, that this is technical talk, but I also would be interested in that entrepreneurial journey of the whole thing, how this success has been achieved. That also would be of interest to us at some point in time. Yeah.
Right. I think we'll ask Geeta to have that journey. I think how we have started, what are the phases.
Yeah, yeah. But she's very busy. I'm not able to get her. Last year also I was trying for the Women's Day thing. So, but
Okay.
I think she was already booked from several places. So,
yeah, right.
So now you do have a patent on this cancer screening.
We have overall 26 international patent, granted patents across the different countries like US, India, Europe, Japan, and Singapore.
Okay.
So this covers different modules of our technology.
Okay. Yeah, and I saw something recently. Yeah, so it has been approved in LinkedIn. I was seeing this is the first one to get approved.
FDA.
Yes.
So we have multiple technologies. Last year, the thermolytics breast screening, which I'll briefly describe in this talk, we got CE approved. So that means that we can take this solution to European markets. This year, a smaller version of it, we submitted for FDA and we got the FDA clearance. So we can sell this solution in US market as well.
Okay, that is the significance of this clearance. So now you will be able to market this in the US. Okay.
Yes.
Okay.
In India, we have been doing it. We got approvals from the Indian government. So we have conducted close to 50,000 screenings all over India. At the moment, we have close to 110 deployments all over India with 19 cities.
Okay. Yeah, this is very impressive. Congratulations to all of you in the team.
Thanks.
Ma'am, can we start now? Ma'am, can we start now?
Yes, Mrinalini can start.
Yes.
Yeah.
Apurni, ma'am, can we start now?
Yeah, sure. Yes, I welcome again for the session 2. I welcome the guest speakers of this session, Akash Narayana and Dr. Shivateja Kakileti from Niramay Technologies. Akash Narayana is a senior data scientist with Niramay and he is passionate about employing AI in healthcare. He has over 2 decades of industry experience in the automotive and healthcare industries. He has predominantly worked on AI and modeling and simulation topics. And the next guest speaker, Dr. Shivateja Kakileti, has over 7 years of experience in AI ML for medical imaging. He is one of the founding members and principal research scientist at Niramay. He was granted a PhD degree by the Faculty of Health, Medicine and Life Sciences of Maastricht University, the Netherlands, and Bachelor's of Technology degree from Indian Institute of Technology, Guwahati. He co-authored 18 international publications, 2 book chapters, and 18 international granted patents. He played a key role in deploying machine learning modules for multiple products that are FDA and CE certified. He was a recipient of DADWISE Fellowship 2014 and was offered Mintax Global Link Fellowship in 2014. This session is going to discuss about the challenges in developing AI solutions for medical imaging, and they are about to present 2 case studies involving the breast cancer detection and COVID-19 detection. Sir, you can start the session.
Thank you for the kind introduction. Thanks, Dr. Chitra and team for inviting us for the talk. Today, I and Akash will be talking about artificial intelligence for medical imaging. Can you see the slides?
Yes, sir.
Okay. So the brief outline of the talk is we'll start with what are the challenges in medical imaging. And Akash will walk you over one case study, which we released last year, which we call it as X-ray Setu, which is an automated COVID detection through WhatsApp. And then I will talk about Niramai's thermometrics breast imaging system and what are the challenges we have faced. And we take one module, how we have evolved our solution. So that is a brief outline of the talk. One of the main challenge in medical imaging is the lack of data. Why there is a lack of data? If you see, the incidence of the diseases are very low. If you're considering breast cancer, on an average, the incidence is 4 in 1,000 women will get breast cancer. So if you want to collect a significant data, we have to screen a lot of people, which is time consuming. At the same time, it is costly also because for every woman, when we are screening, we have to pay their expenses, and then we have to go for get a ground truth. We have to pay for the doctor, all this. At the same time, the data, if not to collect the data, we need to get a lot of approvals. For example, if you want to conduct data collection in some hospital, first we have to take an approval from the hospitals, and then we have to explain the solution. This itself will take around 6 months to 1 year. So once we get the approval, we are allowed to participate in the imaging study. And then depending on the walk-ins, which itself is very low, because even in a top hospital, the number of cancer walk-ins into a hospital would be around 20 per day. That means at most you can collect close to 600 per month. And when we're dealing with artificial intelligence, this data would be low because 600, most of the samples would be negative in nature. And so for that reason, with this low velocity also, the data collection is very tricky in medical imaging. And the next one is variety of data. If we're talking about breast cancer, there are 15 types of cancer, more than 15 types of cancer, including invasiveness of it, whether it is formed in ductal or whether it's formed in lobular, right? So there are a variety of cancers. So when we're collecting the data, when we're training the models, it's important to capture all these varieties, which itself is again a difficult problem because typically any kind of data collection happens in limited hospitals due to the constraints on the money. So for that reason, we will often end up with low variety of data. And when we see many of the open source data collections also, we see all the three problems. The data set, what we get outside is a very low in data set size. There will be usually like 50 images or 100 images, not more than that. And the variety in that data itself also is very low. It will be typically like, if you see some of the X-ray data sets per se, we would see the data sets mostly will consist of X-ray images of children. So that way, the data set variety also, we would end up getting a low variety data set. And the next one is ground truth. Unlike other medical imaging problems, where given an image, any expert can tell whether what is the objects in the image. For medical imaging, it is not straightforward. So if you consider breast cancer, even if you take mammography, the ground truth ultimately is the biopsy. That means the ground truth is not the radiologist annotation, because that itself is subjective in nature. Given an image, X-ray image to a doctor, one doctor might say there is cancer and one doctor might say there is no cancer. And how do you validate this? We take a tissue sample of that abnormality and send it to histopathology. And the histopathology would study the cells and then they say whether they are cancer cells or not. As you can see, this slows down the process of data collection, because now for every patient, we have to send these samples to a histopathology. If we don't take this path, and this is also a costlier path, right? We have to take the samples and we have to send it to histopathology and then get the annotations. If we don't take this path and directly take with the expert annotations, we might often end up with subjectivity in the expert annotations, because even the mammography itself is 90% accurate, right? And when you consider subjectivity, the sensitivity varies from 60% to 90%. So depending on the expertise, the quality of label changes. So it is important to have wide variety of experts when we are getting the ground truth, which often lacks in many of the existing data sets. And the other thing is sometimes we might have to wait for longer periods, especially when we're dealing with treatment kind of problems, right? So the one problem could be we want to know whether a specific treatment is working or not. So that involves following up with the patient for 5 years, 6 years of time, which itself is time consuming. And the other problem is imbalance in the data. That is, as we discussed, the incidence of the cancer is low. That means 4 in 1,000 women for breast cancer will have cancer. That means if you collect 1,000 samples, only 4 people would be positive. So this high imbalance would make the development of artificial intelligence for medical imaging problems a hard problem. The next is choice of ML algorithms. Typically, 2 sets of ML algorithms are being used in medical domain. One is feature-driven algorithms. This involves understanding the medical nature, understanding the domain knowledge. What are the characteristics of cancer from the expert, or taking the statistical samples, and we derive some mathematical features and then train a classifier for classifying whether it is a malignant or not. And there are new sets of algorithms, which we call it as data-driven algorithms, where we don't need this domain knowledge. All we would be needing is the images and the corresponding ground truth. And we expect the artificial intelligence model to automatically learn this knowledge by looking at the images and the corresponding labels. This is a data-driven algorithms. So there are advantages of using feature-driven and data-driven. If you're seeing feature-driven algorithms, they are more interpretable because we're extracting the features and we know what features we have extracted and how they correlate to the output. So one simple feature could be how the tumor boundaries are. Typically, if they are smooth, it corresponds to benign. If they are irregular, it corresponds to malignant. So by looking at the mathematical feature, we can tell, okay, this is malignant because it has high irregularity. It is benign because it has low irregularity value. So that is where feature-driven algorithms are well used when you want to explain your solution. On the other hand, data-driven algorithms, when we see, it is difficult to explain why we have told it is malignant. There is a lot of research that's currently happening by using class activation maps and other techniques. But at the moment, it is still, the current algorithms still are not explainable up to the extent of feature-driven algorithms. But the advantage with data-driven algorithms are that they give very high accuracy when compared to feature-driven algorithms. And the other challenge is deployment of medical imaging solutions. It's not a straightforward deployment that would happen. We can't take a model, or we can't take a solution and directly deploy it. It has to go through multiple steps. This is one innate ML in clinical workflow drawing, which is specified by Kayum in one of the conference. So the first is use case specification. If you are developing a solution, what is its intended use? So the intended usage has to be clear. Even if you want to go for any regulatory approval, you have to explicitly mention who are the user, like who is going to use the solution and what is your solution going to give them. And this use case specification should be very clear. And the next step is when you're collecting the data and when you're training your model, you have to anonymize the data. So any data what you get, you have to de-link with the personally identifiable information. That is a major step which any ML solution has to do. And the next thing is data annotation. That is, once we get the data, we need to annotate this data. So this annotation can be done through passing through multiple experts and getting the label and then picking some kind of average or max or mode of the labels. And other way is to directly go through histopathology kind of ultimate ground truth and get the data annotations. And the next is ML model development. Once we get the data and the corresponding annotations, we will train this machine learning model. This could be feature driven, or this could be data driven algorithms like convolutional neural networks or generative adversarial networks, or any kind of machine learning model. Most of the non-medical use case would end up stopping here and then directly jump to deployment. Whereas for medical imaging, we have 6 more steps. So after the machine learning model is developed, there is an algorithm audit where we have to show that what we are doing is correct, so that we will go to expert clinicians and check with them whatever we are doing, whatever the assumptions we are making, are they correct or not. Now that algorithm audit is done, the next step is showing that it really works. So for that, what we do is we conduct clinical trials. We partner with third parties and use this solution in different hospitals. And we test this, we do something called as prospective blind testing. That is, we give the solution to them, ask them to use it, and we in the end of the study, we collect the labels and we collect the ground truth and compare our results. If this multi-site validation, the results are good, then we have to go through regulatory approvals. This could be CE approval if it is for Europe, it's FDA for US, and DCGI approval for India. Once this is done, we have to release it. We have to have clinical integration. How we can deploy this solution in the current clinical practice, right? There is something called as the current hospitals use PACS system to access the data. So we need to convert our solutions to give the results which can integrate with the existing healthcare workflow. Once that is done, we have to check whether the doctor who is a user, are they comfortable or not. And then there is a real world surveillance. That is, whatever results we are giving, we need to continuously validate them, whether they are correct or not. So, and then this is called as real world surveillance. So once it is completely done, then it will be well accepted. So these are multiple phases that involves in medical imaging. That is why when we see a lot of medical startups and these things, they take a lot of time to deploy a solution in the market, because there are multiple steps we have to do in order to deploy the solution. With this as a background, I will let Akash to speak about the first case study, which is ICCIDS. Akash?
Yeah, thanks, Shivan. You guys can hear me, right? You can hear me, right? Hello.
Yes, sir.
Yes,
Yeah.
sir.
Okay. Yeah. So feel free to ask questions at any point. So Shiva kind of talked about the challenges and 2 key things in the feature-based way of developing the solution. The other was the database solution. So here, Shiva, can you go back to the previous slides? Here I'm talking, we're going to talk about ICCIDS, okay? And ICCIDS is a name given to X-ray-based, COVID detection. And this was something that was done very quickly over the last 1.5 years since the onset of COVID. I mean, it's a very a nice success story, or a story that took place as you know how the world changed the last 2-3 years. COVID came in, it was a quick thing. The world was kind of shaking a lot. And again, if you can look at Shiva's last slide, where he talked about various stages, but sometimes you don't have all that luxury. When something like COVID comes in a country like India, where you have a whole lot of health infrastructure issues. But here is a solution that we did along with ISC and Art Park, which we could quickly deploy, especially in the second wave. So first wave, we kind of developed the model, did some testing, and some of the aspects that Shiva mentioned in the previous slide, checked with radiologists. It was some sort of a soft pilot kind of testing that was deployment that was done. But when the second wave came in, the realization came, look, we need to really scale up. Okay? So this is what you'll see in products. Okay, so can you scale it up? If I come with, you all of us do projects. They're all small academic, but to make it a product, what does it take? And to scale it up to reach thousands of people, can we do that? So that was one of the key challenges out here. So we took an AI model. The AI model remains the same. Almost the AI model is the same that can even handle one. It works for one person. It's good enough. But now, can this AI model give me results for thousands of people in a day? Can I have a system around it? And that was X-ray Setu. So we worked closely with IIC for AI model building. Niramai worked on it quite a bit. We kept the thread for all of these things. And Art Park. Art Park is a non-profit based organization based. Yeah. So I think I have my. Yeah. So Art Park came in and we took the model and we scaled it up. Okay? So that was a very quick thing, that was done in a matter of weeks, and we scaled it up. Okay? So now we'll look at how the solution was made easy so that people could use it, and we wanted the common man, the doctors all over the country to use it. We'll look at the product now. Sure, next one, please. Yeah. So COVID wave 1, wave 2, it was a lot of lung-related issues. We saw a lot of people having lung issues and complications, deaths happening as a result. So the key was to look at COVID pneumonia, as it was called. Can we pick that up? And for that, in cities we have seen more complicated things like RT-PCR and others coming in. But can we look at a more simpler one? That's an X-ray. X-ray is available all over the country. X-rays machines are available all over the country, especially for a country like India, all over, and it can be the results are out, the scan result is out very quickly. Now, can I apply an AI model on that and get the result? So that is the whole idea. But the key thing is, as you see in the slide, I'm not going to read that out, but there has been a lot of people telling that WHO, medical societies, British Society, telling that, hey, do look at X-rays. Okay? So that was the key thing for us. So you can look at X-rays, and based on that, can I do a prediction, or can I kind of estimate, is there a COVID in this person or not? So we can do that, is what these groups said, these publications said. So we used that, and there was a motivation for us to, okay, let's use X-ray based, just X-ray based data, and go in for COVID detection. Yeah, sure. Next one. And I think I covered some of these points. So essentially, yeah. So we'll move on. Sure, we'll move on. Yeah. So how does it work? So I told that we made something very simple. So it is a WhatsApp based a product. So all that you need to do is, it's a free service. You just add the number that is there, and you take a picture of the chest X-ray. You keep it on a white background. Take a photo of it, and just send it. Send that over WhatsApp to that particular number, and then so it goes in, and we have a manual step wherein the technician looks at that image and first checks, is it a chest X-ray or not, and those kind of tests. This was very important for us. I'll come back to that. So the technician looks at it, and perhaps does some sort of sanity check and all that, crops it if required, and sends it to our AI model. So this is the AI thing. So you can see that the AI machine learning stuff that we talk about, we read about, comes here. But there are so many other layers around it to make this a good product. So then it is kind of so this X-ray sent to the AI model, which looks at it and gives a report out. Yeah, we'll look at this. So we'll look at these things, and then the report is sent out quickly as a PDF to the doctor. The doctor opens it and he has an idea. Okay, look, this patient has COVID or not, or what else, and so on. Okay, so this is the flow, and one of the key steps is again the technician who would manually look at the X-ray, and we are trying to automate that step too. But in the last year, her role was very important, right? Because a lot of people were sending non-chest X-ray images too. Yeah, so somebody was sending, pelvic X-ray, or somebody's taking the image, taking the picture of the X-ray very badly so that you get the backgrounds and all that, So imagine keeping it in the back and taking a shot, and I'm sure the crow also would come in the background. So we had images of chest X-ray where we had mountains in the background, so on. So these were the challenges. So and the key thing, the AI model sits there. That is something that we keep on working on, trying to improve. But there are these other layers that make it that are quite important to make it a good and user-friendly product for the doctors. So this was aimed by and large at the doctor. So you have a doctor somewhere, then there's an X-ray, use it, a lab technician, even a lab technician can use it, take a picture, send it, and he quickly gets the report within 30 minutes. In fact, it is even much quicker now. And so the doctor can take a decision quickly. Yeah. Next, please. So the key highlights, as I was mentioning, it's simple and accessible. It's an automated interpretation for COVID patient, and we've seen that the RT-PCR at the height of COVID, there were a lot of delays. They were taking 1 day, 2 days sometimes to get the RT-PCR results. So this was designed so that it can have the prediction quite quickly. And again, you have different types of X-ray machines. It works on both analog and digital. So these are some key things as you build in a model. Okay, so your data should be your model is generalized enough so that it can handle all kinds of data. When I say all kinds, it's more, in this case, it's like analog and digital of a chest X-ray. Okay, so yeah, moving on. So what was now the very key thing in our reports and all that? Yeah, so we went into explainable AI. So Shiva was bringing these two things up, right? He talked about feature-based AI model development, where you can easily say, why this decision was taken. The other is data-driven, so you just give the data completely to AI model. Something happens, it's like a black box. You get an output, but why is that decision taken? Why is COVID there? You're not able to really explain. But there's been a lot of work in deep learning towards explainable AI, and that's what we've used here. So we have gone back and we're trying to say, okay, look. If we are talking about a particular disorder, by the way, we not only mention COVID pneumonia, we also talk about other 14 lung abnormalities. So this AI model has been developed to identify these 15 lung abnormalities. So it tells why is it mentioning this particular abnormality, and how does it say? It just points to that portion in the chest X-ray from which the input has come in. So that's a heat map. A heat map comes up there and say, look, this is what has been, I think, is leading to, say, COVID, or some heart-related things like cardiomegaly and so on. So that's the explainable AI part. This has been, this is quite helpful for a doctor. And the doctor looks at it and he looks at the predictions, and he knows why it has been done. It helps him to make the decision, the next higher level decision easily. Okay. You can say, okay, yeah, true, or you can say, oh, no, this is possibly not possible. And you can rule out a few things, or you can say, yes, I agree with this. So this is quite helpful. Yeah. And coming a little more into the details. So we have the chest X-ray, and now we have a X-ray Setu model, as we call it. So it gives class predictions, as I was mentioning. We have this 15 different lung abnormalities that are given out, like pneumonia, cardiomegaly, effusion, masked nodules. These are all a pulmonologist typically looks at and chest X-ray and comes up with these disorders, finds these disorders. So how is it done? Look, we are not. Yeah, so we'll come to this. We'll talk about that. So as we build a model, so it's still a database model. So the class predictions are given. So there's a training data. So it says, okay, look, this X-ray asks says a cardiomegaly, okay, and annotations in terms of bounding boxes saying, okay, look, this is where we see it. This is what is used to kind of learn used to train the model. So with data like this, we built on it, and the challenges were, all of you are aware, COVID suddenly came from nowhere. So you start with very small data sets, okay? And so how do you go about? How do you so with very small data sets, and even with those, the positive cases probably are quite less, and so with that you have to work on it. We had some open source data, but they were all of patients from a different country. So with Indian patients, so we had to, when we kind of started doing pilot and all that, we started collecting data on Indian patients, and having an explainable layer. So these were the key things that we did. So our approach as we move on, so we learned with different international data sets. We also had an in-house Indian COVID data set that we quickly collected. We had a deep multitask framework that we used to solve these things, and data augmentations. So as I mentioned, the data is quite less. So you want to really augment it up so that we have a much better model. The data augmentation was used, yeah. And then automated, yeah. Okay. So if you look at the model, okay, so when I come at the model, so there is this ResNet 101 layer at the very beginning, which is looking at the complete image, and over the layers, it's kind of capturing, the feature is kind of extracted, in whatever form, And then we had 3 further submodels, okay. So they take these outputs, and one of them looks at, coming up with heat maps, okay. So you have your GradCam based models out here, approaches out here, so where you look at, use those approaches and identify your, lung disease, your COVID-19, put a bonding, come up the heat maps there. And the other one was more of the object localization to tell where it is, So you have bounding boxes up there, and you have again the classification. This was specifically applied to this case of pneumonia and non-pneumonia. Again, the data set matters, So what kind of data we had? So we had data where we had pneumonia, non-pneumonia with bounding boxes. Okay, so let's use this and build up a submodel around here. So we had the region proposal network, the RCNNs to do object localizations, and we had these 2 things. And again, we also had data which is specifically talking about if it's pneumonia, is it viral or bacterial? Okay, so we had one more model to look at that. So we had 2 heat maps, and again we had a separate submodel to look at that. So so this is the broad approach that we used. So as you can see that it is more of deep learning that is used, and the latest architectures and visualizations and stuff that is used. And coming to the data source that we had, so over time we had this. So the NIH 90,000 RSNA, and in-house we just had 260 samples. Okay, so this is in-house when I say in the Indian data set. And so we had still over 130,000 X-ray images. But again, when you look at the the associated labels, you have lung abnormalities for some of them with just pneumonia, bounding box, so on. So it kind of changes. So you have to kind of mix and match and develop these models. Yeah. So we, as I was mentioning, they use GradCam to identify the most contributing pixels. So we use that, and in this case, we had this 14, 15 disorders, right? So it for the ones that were mentioned, it goes and puts a number there. Okay, say 7 is mentioned, I think that legend is missing. So 7 would correspond to a particular disorder. 15 is from this place. 13 is from this place. So this is the kind of report that kind of in the report, a snapshot like this would be there. Yeah. And to look at it, so how does it really compare? So when a radiologist does annotation, he will say, okay, look, I see some particular infection or whatever abnormality out here. And when we look at a heat map, and that is also giving something close to that, So with various the heat map, the temperature, the gradients, and all that. So we're able to really get a localized sense of which are the contributing local regions that are resulting in this decision being made. This is quite helpful for a doctor when he looks at it and when he wants to really understand how these decisions are made. Yeah, and how did our model perform? So on the open source data set, we had 98% sensitivity, 74% specificity, and the ROC AUC was quite good, 0.93. We picked up so this green point is where we had this particular sensitivity and specificity. So this was good, and in addition to this, we were constantly talking, getting feedback, from the field as to how was the performance of it. And on the NIH data set, which is around 90,000 samples, we had again around 0.7 kind of a sensitivity, 7.5 and 7.4 sensitivity, and the ROC was around 0.8. Yeah. Okay, so this is how a sample report looks. This was around kind of 3 pages.
Technology. What are we aiming at? Are we looking at basically nano robots?
Sorry. Okay, I'll continue. Yeah, so this is how a report looks, and where we have, if at all there's a history, we capture that in the findings of it. In this case, it says COVID-19 likely positive. There's a confidence score also mentioned. The lungs, so what is there again? The lungs, the other disorders that are there, like mass, pneumonia, pleural thickening, if it all is there. And so there's a confidence level that is given. And next, yeah. And then we have the X-ray snapshot and the likely places where these interpretations are based on. This is also helpful for the doctor, one, to be also sure that we are corresponding the report is to this particular chest X-ray, because we saw that a doctor or a particular number could send several reports in the same day. So you really need to get back with the report to the right image. So we also have this image snapshot, which helps him to be sure of things. Yeah, and this the third one was with the heat maps, kind of showing for each of those. For example, out here, pneumonia is the top one. So pneumonia, so the heat map shows where is the local the information, where is it coming from. So the GradCam based heat map visualization helped us to do this, and this was quite useful for the doctors. Okay, so I will go to the demo, but in a very okay. Shiva, I think I'll let me summarize and come back to Shiva. Yeah. So in summary, so we launched a free service sometime around April, May of 2021 in a big scale. So report within 30 minutes, and as an AI model, when we validated them, we had a pretty decent score of 0.93 on the use of the COVID-19 data set. And as we mentioned, it not only looks at COVID, it can also be used for other chest disorders. Yeah. So moving forward, as COVID cases come down, it can still be used to look at how the general health or the chest conditions are. And until now, over 9,500 X-rays have been analyzed. Over 4,000 plus unique users have been there. The current third wave that was there, we didn't have. Luckily, it was not so much of a lung-related event, right? It was more in the throat and so on. But the wave 2, we had a whole lot of cases where I still remember a day when we had over 2,000 plus images that came into our system to ICCIDS. So this is what is that we showed that it is possible that marriage of AI and the other services around it. We can give a good service in India and reachable to the common man. Okay, so yeah, what are we doing with time? And,
Akash, do you want to show the demo video which we created maybe towards the end of the talk?
yeah, let's
Prashant has shared that video, right? So we can play that video towards the end, maybe.
Okay, fine. Yeah.
Can I ask you a question
Sure.
at this point in time that it is still not clear to me? You have used the deep learning model like ResNext and other things you have shown. So how did you achieve the explainability?
The explainability is more, that is the GradCam way of doing it. So you have, so when you you have the, what is that? The class activation maps for, so like, let's say we have this 15 disorders. So for each of them, you have a feature map, and that is where you learn it. Okay. So I'm trying to talk about how a class activation map is. So essentially, you have class activations map, which are defined for each of these, and we use GradCam, that's gradient based visualization. So the CAM for support was much more simplistic. You have you know the output, you know the output, and you try to go back and see from where the most contributing pixels or the weights are coming in. So essentially, that is the idea. So you have those feature maps for each of these, and again a gradient based weightage for each of them. And when you sum that up, you clearly get a heat map showing which are the areas that have been contributed more for addition making.
Just to add what Akash is saying, Dr. Chitra, we have two explainabilities. One is, as Akash is alluding, is explainability based on radiological findings. The 14 classes which Akash has presented, the doctors call them as radiological findings. That is, these are the features we can say. In other words, we can say these are features. Based on these features alone, they can tell whether they have tuberculosis or whether they have any other radiological disease. So what we did was the first level of explainability we provided. Instead of just saying whether the patient is COVID or not, we also told them what are the radiological findings, whether there is a consolidation, whether there is infiltration, these kind of radiological findings. These radiological findings sum up to the radiological disease such as COVID-19. So that is one layer of explainability we gave. So when they see whether they're COVID-19 or not, they can also see, okay, is there a consolidation, which is a main feature of COVID-19. So they can validate it. So that way, that is one kind of explainability which X-ray Setu has provided. The other is localization. When we say COVID-19, we just don't give just probability and then leave it. We localize where the algorithm thinks it is COVID-19 based on gradient class activation map. Now the doctor can go there and then see at those portions and then see if there is really anything there or not. So that is the second level of explanation. With this, both the explainability, it is more easy for a doctor to interpret the image instead of just simply saying whether the image is COVID positive or not.
Okay, thank you, Shiva. Yeah.
Okay. Any further questions? Okay, so if not, I think yeah.
Good morning, sir.
Yeah, go ahead.
Thanks, Akash. So I would continue with this on the next case study, which is Niramai's thermolytics breast cancer screening, which is a main product of Niramai.
I think someone else wanted to ask a question, I guess. Kavita?
Oh, okay.
Ma'am, this is Minu. I have a small doubt. So here in that prototype, what you have shown, this WhatsApp application is used for getting the image and then sending the report. The actual work is actually done by the analyst. Am I right? Not in the mobile platform. The deep learning concepts. WhatsApp is being used as an getting the data and finally sending the report to the doctor and finally to the patient. Am I right, sir?
Yeah, you're right. In the sense, WhatsApp is, Shiva, I'll just share the screen and show the WhatsApp page, and then you can also answer that. No, can you stop sharing? Yeah. Can you see my screen?
Yes, sir.
Okay. So this is X-ray Setu. I'll just quickly show how things are happening here. Okay. So what happens is, let me go. So users starts by adding this number and sending an hello a message. So they get a report. They get a message like this saying how to do it, how to use it, and so on. And say further on, I'm just using what has been sent earlier. So you send a message, X-ray image like this. You get back a report like this. I'll answer your question. Okay. So you get back a report. So you get back a report. Yeah. So on WhatsApp it goes. So we have other services which will transfer it to another system or to a distributed system. In our case, an Amazon-based server, which will where all this communication happens. Okay. So yeah, when I say communication, so when you send a message on WhatsApp, so there is let's say a cloud service which will pick that particular, we have APIs which will pick up that image. Then it goes to a technician. A technician looks at it and says, okay, look, this is good or not. I want to go back to my. Okay, I think I lost my WhatsApp.
Fine, sir. So it will. What will be the turnaround time?
Turnaround time could be as quick as even 3 minutes, 4 minutes. If you have a technician who's working on it right away, it is like a few minutes, but our SLA is within 30 minutes. So we have a bunch of technicians available on the other end. So even if tens of X-rays or maybe even let's say hundreds of X-rays come in an hour, we are able to handle that within 30 minutes. So the model computation time is almost nothing. So essentially the time that goes is when the technician manually opens it, just make sure if it is fine, if they have to crop it or give some other reply. Say, okay, look, this is a wrong image and send it back. So the moment they think it is fine and they again say submit, it goes back to our model service on AWS, and that's just a matter of seconds. So our average time, I think in the last few months have been, even sometimes even to go down to 4 minutes, 5 minutes, and so on.
Okay.
Okay, great. Yeah.
Sir, I have one question, sir. You will be explaining about the explainable AI, no, sir? From the heat map, you are going to give the explanations how the predictions has been carried out.
Yeah.
Yeah, actually, currently we'll be having many libraries or packages with supports Python for explainable AI, no, so that is SHAP, Lime, explainable dashboard, like that. We'll be having some packages, no, sir. Are you using any of the packages, or you'll be doing the prediction from the heat map itself, sir?
No, from the heat map only, it was more of.
Okay, even this packages also supports with heat map only, no, sir?
Yeah, but I think, sure, you want to add the implementation, I'm not very sure. I don't think there's a need for it because, we probably would have used just a GradCam and because, see, we need to just visualize the, the key pixels for a particular in a class, right? So that is all we do, we are not going beyond that. So that is sufficient for a doctor to say, look, if I say COVID, this particular area of the lung is where it has been used to make this decision. So this CRAT-CAM kind of an output, visualized output, is good enough for us to do that. Not other classifications were in the other submodels.
Okay, so it is not mandatory that we have to use only the explainable AI packages for that task. Even the heat map itself is fine, no, sir?
Yes, heat map is part of explainable AI, right? I mean, it's part of your visualization.
Yeah.
So that gives the heat map. So once you have the heat map, you're done actually.
Okay, thank you, sir.
Okay. Shiva, I'll do that.
Yeah, can you see the screen?
Yeah.
Oh. Yeah. So just a brief introduction about the breast cancer. It is one of the leading causes of cancer deaths among women. And if you see the statistics, 627,000 women lost their lives across the world in 2018. Those survival rates in developed countries like India is 80%, sorry, US is 80%. Developing countries like India, it is only 50%. That is, every 2 women diagnosed with breast cancer, 1 woman lose their lives. This is mainly due to the late detection of cancer. Often in India, when women is diagnosed with breast cancer, they are in stage 3, stage 4. So the chance of survival is very less. So what are the main reasons for late detection of cancer? If we see, conventionally, X-ray mammography, ultrasound, and MRI are used. X-ray mammography is a gold standard. However, it is applicable for women above 40 years because of the dense tissue issue. How X-ray mammography works is, if there is a cancer, it has high tissue density. So based on this difference, it tries to find whether there is a cancer or not. But for younger women, that is women less than 40 years, the tissue itself is dense. So it is very difficult for the radiologist to separate between malignant tumors as well as normal dense tissue. That is what is recommended for above 40 years. Other reason is X-ray mammography use radiation. So if you use frequent X-ray mammography, even though you don't have cancer, the chance of getting cancer doubles after 10 screenings. So for this reason also, even different breast cancer programs, they typically recommend X-ray mammography only once in 2 years or once in 3 years. But the main reason why in India X-ray mammography is not so popular is the equipment cost. The cost of the equipment is very high and it is infrastructure dependent. It has to have a separate room with concrete walls, and also it requires manual expertise, both in image capture as well as image interpretation. And final reason is the technician have to the patient has to disrobe in front of a technician who often turns out to be a male technician. So due to this privacy issue also, many people don't prefer X-ray mammography. Ultrasound and MRI are used, but in they substitute X-ray mammography. That is, along with X-ray mammography, these two are used. So what in Ramai we are trying to do, can we come up with a solution which is completely non-invasive, privacy aware, and comfortable for the patient? So that's when we looked at breast thermography. So breast thermography involves the measure of temperature values emitted from breast surface. What happens when there's a cancer, right? Cancer is nothing but uncontrolled cellular growth. So when this happens, for this cell division, it requires more resources. So this the resources will be supplied using blood supply, using angiogenesis, which is formation of blood vessels, and dilating the existing blood vessels to pump more blood, and also recruiting the dormant blood vessels to get more resources. All this activity that's happening in the vicinity of tumor generates heat, which will be conducted to the surface through venous convection and tissue conduction. And at the moment, the thermal cameras which we have, they can detect temperature variations up to 0.02 degrees centigrade. So it is possible to study the heat variations due to this abnormal metabolic activity. So with this, we can make the breast cancer screening non-contact and non-invasive, because what we are essentially doing is capturing the heat that's emitting from the breast surface. So we don't have to touch the patient. There will be a thermal camera, which I think most of you might have seen currently in airports or office spaces. So there's a thermal camera which will be like 3-4 feet away from the patient, and we capture the heat that's emitting from the body. So it's completely non-contact and non-invasive. Since we are collecting only the radiation emitted from the body, which is not harmful, it is radiation free. It is a portable, light, small, low-cost imaging device. This we can carry even in a backpack, very small device. And we can make the entire screening privacy aware. So what we can do is, typically we place the camera in a room, and we tell the patient to go inside a room, or we have a cloth partition, and we control the camera from outside, so that even the technician who is imaging need not see the undressed woman. And it works for women with dense breast, which is an issue with the current gold standard X-ray mammography. And the challenges with manual interpretation, right? So if you see this image, what thermal images captures is raw temperature values. So in order to view, we have to convert this into some kind of color images. So typically what thermologists do is that they have one temperature lower value and higher temperature limit. With this, they use a color palette like this, which is called rainbow color palette, and maps these temperature values linearly across this palette. But as you can see, if you change these limits, the temperature pattern has changed. So if you have set 25 degrees centigrade and 39 degrees centigrade, we are seeing no tumor at all, no hot patterns. But when you keep 28 to 36, you can clearly see there is a tumor. So this causes a lot of subjectivity in interpretation, and it requires high expertise to understand and study these heat patterns, as well as these heat patterns can be caused due to benign conditions. So we need to know what are the main features that contribute to malignancy. And that's where the expertise of thermologist comes in. So what we have done at Niramai is we developed an AI-based solution which takes this thermal image and automatically generates a report which can help the thermologist or thermographer in interpreting the breast thermal image. In order to do this, we need to have multiple machine learning models. First, we need to ensure whether the image is good or bad enough for processing. So what happens would be many times the image is not taken properly. So one simple error could be focus shape. So when we are capturing these images, temperature matrices, the camera might not be focused well. So the temperature values would get averaged. So we have a machine learning model which will automatically tell whether there is a focus error or not. Another thing is for breast thermography, we capture 5 different views so that we cover the 180 degree of breast region. So we capture something called as frontal view, side view, and complete lateral view. Similarly for left and right. When we capture this, one of the common errors that happens is that technician label left image as right image, because when they capture the image, on the left hand side, there'll be right breast, on the right hand side, there'll be left breast. So due to this confusion, they typically label these images wrongly. And when they upload this, the algorithm also might give faulty results. To avoid that, what we have is auto labeling or auto tagging algorithm, which given an image, it automatically tells whether it's a side view or a lateral view or a frontal view, or whether it's a left view or a right view. Once it is done, the next step is region of interest segmentation. The thermal image, since it is capturing from far, along with breast region, it captures the heat pattern that's emitting from the neck, abdomen, hands, all these regions, right? Since we are interested in only in the tumors that are growing in the breast region, we have to segment the region of interest. So we have a deep learning model to segment the breast region. The next step is there is a tumor localization, that is in the breast, where is the tumor or where is the vascularity. Once this is done, we went with the feature-driven approach. That is from these tumors and vascularity, we have extracted using domain knowledge and statistical knowledge, we have extracted some features which correlates to the malignancy. So we have one final classifier which takes these features and classifies it as malignancy or not. Further, if it is malignant, we have one more classifier which tells whether the cancer is due to hormonal in nature or whether it is hormonal negative. This is the overall pipeline that we use for detecting the breast cancer. In this talk, I will talk about one component, which is region of interest segmentation, how the model has evolved over the time. So I'll introduce the problem. I will explain how and when we used feature-driven image processing approach. And then we go to data-driven approach. And then we discuss merits and limits of both the approaches. So these are the 5 images which we capture for breast thermography. So this is called frontal view, and this is left lateral, this is left oblique, and right lateral view. As you can see, unlike standard breast segment object detection algorithms, there are no strong boundaries. There is no strong upper boundary. Those side boundaries are strong. There is no upper boundary or side boundary here. So for this region, even when we ask an expert to segment the breast region, the DICE index or the similarity between the region to the segment, it's only 91% accurate. That means even the best algorithm can't give more than that, because there is a subjectivity in the ground truth itself. So the problem here is it's not only one view, we have 5 views. So we need to have an algorithm which can work on multiple views. We don't have strict boundaries like other object detection algorithms. And inter-observer DICE similarity is only 90%, which is subjectivity in ground truth. And when we started this problem, we had very less data set. We had only 50 subjects, and which is taken in only one hospital. So it is of low volume as well as low variety of data. So we can't directly develop a model with this and release it and be confident that it could work for all the cases. So what we have done is we went with traditional feature-driven approach. So we understood, we have come up with heuristics. What features we can use to segment this breast region? So we thought the breast region is nothing but it's a region that's enclosed by 4 boundaries, which we call it as lower boundary, upper boundary, side boundary, and mid boundary. The lower boundary which you are seeing is formed from inframammary fold. The side boundary, what you're seeing here, is formed from body boundary. And the other side boundary which you're seeing is formed from sternum. And upper boundary is formed, there is no strong boundary. So we have to join this with some heuristics. So first, as a first step, we thought of segmenting the lower boundary because it might be a simpler problem. So what we're seeing here, this high heat pattern, it's always caused because of the inframammary fold. That is, because of the sagging of the breast, we always see friction between the breast and abdomen region, and this region is hotter. So what we have done is we have taken a histogram-based approach, and we have come up with some thresholds depending on the body histogram to identify the high temperature regions. Once we get the high temperature regions, we can take the upper boundary of that temperature. We can take the longest region and take the upper boundary of this region to get the lower boundary. So that's how we got the lower boundary. We can go with edge-based detection techniques as well, but the advantage with region-based techniques is that it will give us a continuous border. So that's why we went with the region-based segmentation technique. And to get the upper boundary, what we have done was we tried to segment axillary region. Axillary region is the hot temperature region. So we try to segment the axilla hotspot, and we have taken the lower boundary of that axillary region as the upper boundary of the breast region. This is one approach which we have tried. And to get the axillary region also, we have taken this histogram approach and considered the longest hotspot in the top 1/3 of the image. But what happened is some cases we are not, some close to 50% of the cases, we are not seeing a high temperature region, the axillary region, like in this case. So for those cases, what we have done was we had went with heuristic. The heuristic is that we have assumed the breast height as half the abdomen height. In order to get the abdomen height, we first segmented the body region using auto segmentation. So auto segmentation is nothing but the threshold at which we can segregate the cluster of foreground and background. Once we get the threshold, we can get the foreground region, and we found the width here A, and then we have taken a straight parallel line which is above A by 2 here. So that's how we got the upper boundary. This has worked for most of the cases and gave a very good results compared to the approach one. And to get the side boundaries, we have taken, once we get the foreground region, we simply applied any edge-based detection technique using Sobel operator or Pointed operator. So, and we got the side boundaries. And for the mid-body boundary, again, we went with heuristics. If it is a frontal view, because both the breasts are symmetrical, we assumed the mid-body boundary is a line that segments the side boundaries in 1:1 ratio. And this happens to be same for even left lateral view or the right lateral view. For an oblique view, which is a 45-degree view, what we have found is that the breast regions which we see would be close to 1/3. Only the other breast is around 1/3 region. So because of that, we went with an heuristic of 1:2 ratio to get the oblique view boundary. Now, with these 4 different boundaries, we can just overlap this and get the region that is enclosed between them. So this is how we got the breast segmentation. So in order to measure our algorithm, what we have done was we try to go with dice index. So in this scenario, accuracy might not be a good metric. As you can see in the entire image, only this portion is breast region, and almost 75% is non-breast area. So when we go with metrics like accuracy, they give false result, because even if you say everything as empty, that is negative, we still get 70%, 80% accuracy. This happens because of the imbalance in the positive and negative samples. So for this region, we went with dice index, which is a similarity metric, which tries to measure what is the overlap between the predicted region and the actual ground truth. When we tested on a small data set, we got 80% accuracy. However, the main issues with this approach is we have used multiple heuristics. And these heuristics are dependent on how the technician captures the images. If the protocol is not followed, the segmentation might not be accurate. So when we tested in real world scenario, the results dropped to 70%, 65%, because the imaging protocol we are not able to ensure. So the results are highly dependent on the imaging protocol due to the use of low-level features, what we have used. So that when we went with hybrid intelligence, that is, instead of saying that we will take the region that is predicted by this approach, we said that we will use this approach as an initial precursor for the breast region. So whatever region we got, we have displayed in Polygon user interface. And this will be displayed as soon as the images are uploaded to the technician. And technicians can drag the boundaries in case if it's required. And if it is proper, they can simply go next. So this way, what happens would be it speeds up the segmentation. If you ask the technician, human expert, to draw the breast region, it takes a lot of time. But with this, they have to adjust only a few points, and then they can quickly go to the next one. So what we have observed is that it has increased the overall, decreased the overall time for segmentation, and has improved the number of images that are captured by the technician. With this initial setup, we got some 1,000 subjects. That's when we switched to deep learning. So deep learning is a data-driven approach, which, as discussed, has shown very significant results when compared to feature-driven approaches. But the main challenge here is we need to have sufficient data. So since we're dealing with images, we go with convolutional neural networks. So what convolutional neural network does is that instead of relying on manual feature engineering, that is how we have extracted the low-level heuristics like the upper boundary of inframemory fold, side boundaries like that. It looking at the images and the corresponding ground truth, it abstracts these features automatically. So, for example, this is a classification approach problem. So, if it is the feature-driven approach would be something like how many strokes are there, right? If it is 1, there'll be single stroke. And if it is 9, you will see some kind of circular structure. So like that, we used to have heuristics in order to identify the digits. But now what convolutional neural networks does is it takes these images and classifies them into direct digits, right? So we have more convolution layers, which takes this, abstracts this input, and then forwards to the multiple layers to take a final decision whether it is which digit. So for this deep learning problem of breast segmentation, there are 3 main steps. One is we have to identify what type of model architecture we have to use, and what is the training data, and what is the loss function. These are the 3 steps we need to know before we train any deep learning model. So for the model selection, we went with VNet architecture. VNet architecture is a variant of UNet architecture, which is an encoder-decoder architecture. What it does is, if you have image of some higher dimensions, it downscales the image to extract high-level information. And from this latent variable dimension, it again upscales the image to reconstruct the final output. And at each and every stage, the encoder and decoder is connected so that while upscaling, it can take this information and upscale it to be similar to the input image, to take the information on the input image. And this UNet or VNet architecture, they have shown very good results when dealing with medical images. The next one is training data. So any training of deep learning model requires a lot of data, but we only had 1,000 subject data, which contributed to 5,000 images. So the question was, would this be sufficient to train the deep learning architectures? Which definitely this VNet architecture has millions of parameters to train. And when we have only 5,000 images, the architecture tends to overfit to the training data. So what we have done was we have performed data augmentation. So one augmentation we have done is Gaussian noise. So as you can see, this Gaussian noise we can assume as some kind of interference that might happen during images, right? So depending on the humidity, depending on the dust or dirt particles during image capture, there will be some kind of noise added to the images. So to simulate those kind of scenarios, we have added Gaussian noise. And then we have created multiple images. And random crop and resize is another operation which we have applied. Because the imaging is subjective in nature, some technicians might take from the lips to abdomen, some might take from neck region to abdomen, and some might take a little bit above breast region to abdomen region. To simulate these kind of scenarios and to make our algorithm more agnostic to these variations, we applied this random crop and resize, where we automatically cut some portions from top, bottoms, and side to simulate these scenarios. And random region insertion is one operation which we have done. So what it does is, because there will be more negative samples compared to positive samples, in order to simulate malignant cases, what we have done was we have taken some of the regions inside the breast and then manually increased and then randomly increase the heat pattern. So that way they form some kind of hotspots in the breast region. So this we try to simulate in order to generate positive kind of cases. And finally, thermal sensitivity modification. In the market, there are multiple thermal cameras which are of different resolution. So when we are developing a solution, we have to make sure that this works for different sensitivities. So for that, we have taken the highest resolution camera images, and then we simulated the images which corresponds to low sensitivities. For all these augmentations, we have considered flipped images because that is also a valid image where the right breast is left breast and left breast will be right breast. So with this kind of augmentations, we have got millions of data. And the next step is ground truth ambiguity. Here, as you can see, if it is a frontal view, we have to segment 2 regions of interest. And if it is an oblique view, we have to segment only one breast. So how do you learn, how do you tell the architecture, to segment only the dominant breast, even the other breast is visible? So for that, what we have done was we train the architecture such a way that it only detects left breast in the image. Even the right breast is visible, it will not detect. So for a frontal image, it will only segment the left breast. Now, in order to get the right breast, what we do is we flip the image and pass it. So that way we avoided this ground truth ambiguity. And instead of asking it to automatically figure out whether to segment 2 or 1 breast, we told always segment the left breast in the image. And for the loss function, we had bandwidth dice loss, as I earlier discussed. Accuracy might not be a good metric here because there is a lot of imbalance between positive and negative samples. So we have taken the dice loss and we have modified for our use case. So the dice loss here is represented by 2 into summation of pi gi and summation of pi square plus summation of gi square. Along with this, we have added one more term, which is nothing but distance between the centroids of the predicted region and the ground truth. Because in this scenario, we are 100% confident that for an image, there is only one breast to be detected. We said that the centroids have to be as close as possible. This constraint why we kept is that even though we predict little bit outer region, that is still fine because as we discussed, even for an expert, it is difficult to segment. There is no hard upper boundary. So even though slight region that is above this boundary is detected, it is still fine. So for that, we have added the centroid, which ensures that even if you predict a little bit above, the centroid might be still the same. So with this kind of training, as you can see here, the results, what the blue regions, what you are seeing is the region that are segmented by the algorithm, whereas green is from the manual segmentation and aqua region is the overlap. Irrespective of the imaging protocol, that is whether the image is too far or whether the image is too close or the camera is too low, in all these scenarios, we can see the segmentation that is predicted by the algorithm is very close to human segmentation. Whereas when we go with manual segmentation, the traditional feature-driven approach, the results are not great because in this scenario, it is not exactly 45 degrees. So it is close to 55, 60 degrees. And as you can see, the half portion of the breast is cut. And in this scenario, this is a thermal image of younger women. So there is no breast sagging, there is no inframammary fold, because of which the inframammary boundary is not detected well and the breast region is improper. So the deep learning wise, it gave very good results irrespective of these scenarios that happens. And when we compare the results, the dissimilarity is 92%. This is almost similar to the inter-observer dice correlation, showing that the proposed solution performs similar to human expert. It is robust to image captures. And to answer the question whether the data that is sufficient, or will our results improve with the data? So clearly the results wise, if you see, we are getting 91, 92% dice similarity, which is close to the inter-observer dice correlation. So that shows that this might be the maximum accuracy we might get. And to verify that, what we have done is we have trained the architecture by taking the data set and then by training with only sub portions of the data. So we trained with some close to some 10, 20 images, the deep learning architecture. Then we found 55% accuracy. We increased the data set to 500. Then we found close to dice index of 70% on the test data set. And we increased it further. As you can see, after 10,000 images, the dice index almost remained constant. This shows that even with more and more data you add of similar distribution, we would end up having similar dice index. So that was the result of this experimentation, because this is one question which we get, whether adding more data would help to our model or not. So as you can see in this scenario, it has helped up to a certain extent, but after that, it remained almost same. So in order to get more results, either we had to add a different variety or it might not be possible in this scenario. So the next step, essentially when we are in this phase, right, where the amount of data is low. So we have to simulate more data, or we have to get variety of data. So one way we discussed is data augmentation. But the problem with data augmentation is it would end up giving similar training distribution, but we need to have variety of data. So for that, we need more complex architectures. So that's when we have pursued generative adversarial networks. Briefly, talking about generative adversarial network, there is a generator architecture and then there is a discriminator architecture. So what generator architecture does is it takes Gaussian noise as an input, some random noise, and then it generates some random image. Now the discriminator job is to identify whether it is, for example, in this scenario, digit or not, whether it is a valid digit or not. Whereas generator objective is to generate the images that are close to the digit. So how we formulate this problem is, first generator takes a random image, generates a random image by taking a random input. Now, the discriminator is asked to classify this image as a fake image or this image as a non-digit image. So to do that, we train the discriminator by giving the gi, xi as input, where here gi is the generated image and xi is the actual input image. And obtain the loss or train the network by assuming ground truth as 0, 1. In the next step, what we do is we train the generator network and we freeze the weights of discriminator, that is discriminator is constant, the network weights. And then we ask the generator to generate the images so that the discriminator will predict them as 1 or ground truth as 1. In other words, it can be formulated as a two-player minimax problem where we try to maximize the expectation of the discriminator matrix and minimize the generated error. So we have taken this generator adversarial network approach and then we have applied for best thermal images. So what we have done was we have taken a Gaussian noise as an input, and the generator is generating some random images. And for a discriminator image, during discriminator training, we told that whatever generator generates, it is a non-breast thermal image, and these are the breast thermal images. So the discriminator is trained well to differentiate between these two. And some steps, or during some training steps, what we have done is we lock the discriminator weights and train the generator weights such a way that discriminator will tell whatever image that is generated by generator as breast thermal image. Now, this two-player minimax game will continue for some epochs, and at some point, generator generates good images very close to the actual distribution. So this is one generator output after 150 epochs. And these are some of the generated images from the from the current approach, what we said. So none of these images are in the training set.
But he wanted the other person to give some feedback. And it comes today, let us get the feedback. And இது வந்து ஒரு ரிவைஸ்டா ஒரு டெட்லைன் சொல்லிக்கலாம். இது இருந்ததுங்கிறது ஆல்ரெடி சொல்லியிருக்கோம் இந்த ஹேக்கத்தான். அதனால
Yes, sir. Sorry.
As you can see, all these images are not in the training set or any kind of test set. All of these images are randomly generated. Now, once we have these kind of images, we can pass through experts and get the segmentations and use this data for training different models. So this is one other way to generate complex augmentations to train the models. So the other one, just a second. Yeah, sorry. And finally, to conclude, the other way of improving is on-the-go learning or active learning, where you can have the model deployed, and then you can have this polygon approximation and then see how many times the user has adjusted, find the error between this approximation and user refinement. And whenever you see there is a lot of refinement that's required, you can train the model with the new samples. This kind of active learning approach could also help in making your architectures learn on the go. Thank you.
Participants, if you have any queries, you can ask. Sir, I have a question.
Yeah.
Regarding with the thermal images, I hope that these images are not most probably used in any of the hospitals or something. Only few hospitals may be using this one. So
Right.
for that one only you have discussed about the augmentation technique. But when we ever use an augmentation technique in a deep learning model, we didn't know how many images have been generated by the augmentation technique. Is it possible for us to know or to view the images that have been generated by the augmentation technique?
So you're talking about open source data sets. How do you ensure whether some of the images are augmented or not?
Yes.
So in most scenarios, it would be mentioned in the open source data set itself if they are using any kind of augmentations.
Okay.
But what I have seen in most of the data sets is that when they open source the data set, they will explicitly mention that they have performed some augmentation and generated. But in most scenarios, they do not do augmentations because augmentations unnecessarily increase the data set size, and they have to bear the storage costs as well. For that reason, so what they give is they give the data and they give a function which will generate augmentations. That's what I have most seen.
Okay. But if you are looking onto this one, I am taking a particular image and I'm applying any transformations, augmentation functions with most probably all the transformations we can apply onto the image. So if you do that one, will it be a useful thing? Will it give me a better model if I use augmentation images for building my model?
Yes. So two reasons for it. As we have seen, right, the augmentations which are mentioned specifically for thermal images, we talked about Gaussian noise addition. So what Gaussian noise addition is helping is that if there is any kind of noise that happens, you're already telling the network that these might be the noise that might happen during the image capture, and you still have to predict this. So you're already making sure that the network is invariant to this kind of noise. Similarly, the technician variations will come, right? We will not be able to capture different technicians, how they're capturing. So for that, we kept random resize and crop. Similarly, thermal sensitivity. So all these augmentations, what we have applied had a reason that we're making sure that algorithm is robust to these variations. So one is variations angle. So even if there are some kind of variations to the images, the algorithm should still predict the same output. The second one is on if you're using generative adversarial network and to generate complex architecture, complex images, or more sophisticated images. The training, when it's learning, it would learn better. So in both the scenarios, we have conducted one small experimentation where with augmentation, what is results, and without augmentation, what are the results. And what we have seen is that there's a close to 5 to 10% improvement by using augmentation.
Okay, thank you, sir. Participants, is there any question?
I think one question is mostly on is there open source? The dataset can be open sourced, I think. So the dataset we have collected at Miramai are proprietary in the sense that we have signed an agreement that we would not be sharing it with outside. But there are other thermal data sets you can get from Visual BR. You can search for Visual. I'm just typing the chart. Visual BR. You can search for this lab and you would be getting for breast thermal images. And we have open sourced, not for breast cancer, for other data sets. And this is on, one is fever test. That is from the thermal images, can we identify whether the patient is wearing mask or whether the person is wearing mask or not, or whether the person has high temperature or not. So that is one data set which we have open sourced. The other data set which we have open sourced is OncoCircaSys data set, which is from the heat pattern, can we tell whether there is an oncoworm? It is one of a prominent disease in African countries. So those two data sets we have released. Maybe afterwards I will share both the links of this data set so that some of you can try this. And we are also conducting one workshop, not workshop, it's a conference in medical imaging computing, MICCAI, which I would be sharing with maybe Dr. Chitra. And you can use these data sets and you're welcome to submit papers. And MICCAI is, as many of you know, it is the top tier medical imaging conference where this year Niramai is hosting medical imaging with thermography conference. So you can use these workshops and you can plan to submit papers there. I will share more details with maybe Dr. Chitra afterwards.
Thank you, sir. Thank you for your information.
Yeah.
I would like to request the participant to switch on your video so that we can have a photo session now.
There's one more question by Kavita. Name of heuristic-based segmentation. Sure, I'll just try to answer that. See, you want to add on. So that's a good question. Okay, when you look at heuristics, you wonder, is there a rule there? Is there something somewhere in some textbook? But it's not really that way. When you're trying to solve a particular problem, okay, in this case, breast images, say, so the solutions are very specific to that particular problem that we're solving. Okay, so we come with our more of some common sense based approaches to solve it. So it's very specific to what Shiva showed in the last 40 minutes, 45 minutes. A lot of them are very common sense or well thought out approaches to solve them. So sometimes you will not really find it in the literature, but this is how you solve it. You look at literature, then you really look at the data and see how really you would solve it, and you come with your own heuristics.
So the work that in which you have used deep learning models, can you list out the deep learning models you have used in your thermal imaging, both for the segmentation and localization?
So some of the architectures which we have used for segmentation per se, if you are saying variants of UNet or encoder decoder architectures, they perform well. So it's called UNet or VNet. These are encoder decoder architecture for segmentation. Whereas for classifications, what we have found is BGGNet and ResNet architectures, they perform well.
Okay. Yes, participants are requested to switch on their video. We have a photo session. I kindly request the participant to switch on your videos. It's any more questions? I think we came to the end of the session. I thank you both for your talk on artificial intelligence on medical imaging. You first start with the major challenges that is prevailing in the medical image processing, the implementation of the X-rays. X-ray Sedu gave us the insights of the explainable AI. The importance of burst terminology was also discussed, and that introduced us a new modality. The talk was inspired many of us to explore in this new area too. I would like to thank Dr. Chitra Babu, who helps us to get connect with you. I thank once again for both of you for sharing your valuable time with us. Thank you, sir.
Thank you for inviting us. It was great meeting you all.
Yes, we are looking forward to interact with you for future sessions too. Thank you.
Thanks everyone. It was a pleasure. So I hope it inspires some of you to pick up some interesting problems. Thank you.
Yes, sir. We saw many papers. We didn't have any practical implementations or practical persons who worked on it. We didn't never had a talk with them. So I hope that people who are working in medical image processing, your talk gave them a very good startup or motivation that we can also do work in medical image processing too. Thank you, sir.
Thank you.
Thank you, sir.
The feedback link has been posted in the chat box. I request everyone to give your feedback on this particular session, on the second session. And now we will break for the lunch and we'll meet at 1 o'clock for our afternoon session. Thank you. be awake here, and it will try to compute the dot product to get the convolved feature max. So the dot product that is obtained after adding the bias and then after giving it to an activation function, I'll get a feature matrix. So that is what, so this is what is my input image. This picture shows the convolution operations. So this is what is the input image, and this is what is the filter. So it is one filter. Similarly, the convolution layers have n such filters that is present. Each filter helps us to extract one particular features from my input images. So this filter will keep sliding from the leftmost corner of my image to the rightmost corner of my image. So the first time when I keep this the size of my image is 7x7, and my size of my filter is 3x3. So I will try to slide each and every position of from the each and every position, and I need to determine the stride position also. So how will I am going to stride? I am going to stride each and every pixel, and or I am going to stride 2 pixels once. So this is what is said to be the stride. So I need to determine that one also. So I'll keep this filter onto the image and find out the dot product. That is what has been obtained as my feature map. So this 0 is been obtained by convolution operation of this filter onto this image. So this is what is the feature map that I obtained by with the help of my filter 3x3 onto my image 7x7. So the first thing has been the filters have been slided. They slided along the over the depth and height of the input volume. So it will produce a 2-dimensional feature map or an activation map that gives this is nothing but the responses that I have taken from every spatial position of my image. So I will not have one such filters. I may have n such filters, where each filters will extract one particular features from my images. So this is an image where it consists of four 3x3 filters, and each has generated some part. So each will produce 2-dimensional activation map. All these activation map will be padded in order to produce the feature vectors. So this activation maps are stacked along the depth dimensions, and it produces the output volume. Once this feature map has been created, this have been given to a non-linearity activation function, such as which is called to be a ReLU here. As I told you, the earlier layers, that is the input layers and the hidden layers, will have a ReLU as an activation function. So that is what the stacked feature maps will be given to the activation function. So now I want to discuss you something which is what is called to be the hyperparameters. So what do you mean by hyperparameters? The hyperparameters are nothing but if we keep tuning the hyperparameters, I'll get a better accuracy in case of my any classification problem. So what are the hyperparameters? Normally these are the 3 hyperparameters which is normally used in any deep learning algorithms. Apart from it, we have many other hyperparameters such as number of epochs, or what is my number of images that have been taken for consideration. Everything is considered to be the hyperparameters, but fine tuning of these hyperparameters will help us to get a more perfect results. But how will you fine tune it? This which hyperparameters need to be fine tuned? It all been decided based upon what is the problem and what is our input, and what is we need from the input. So all these decides upon this one, and what is most probably the hyperparameters that have been considered in case of my convolution layers is nothing but the depth of the output volume. So how can I say decide onto my depth? Whenever the number of filters keeps on increasing, my depth of my volume will keeps on increasing here. So the number of filters decides the depth of my output volume. So this it depends on the number of learnable filters I am using onto my input image. And the next one is the way in which it has to be slided. So how I am going to do the sliding operations? Is my slide is equal to 1? If my slide is equal to 1, I am not going to miss any one of the pixel. I will slide each and every pixel starting from the top left corner of my image to the rightmost corner of my image. So that is what is stride. So when the stride is 1, we move filters 1 pixel at a time. When the stride is 2, we skips 1 pixels and jumps 2 pixels at a time. So this is what is said to be the striding. So that striding is also another hyperparameter which decides on the accuracy. And then the next part, which is what is called to be padding. So when when you look onto this image, I keep striding each and every pen wherever my stride is equal to 1. I'll start with this leftmost corner and get the product of this one. I'll move my thing and get this product, move this one. When you move on to this one, what will happen? My filter will be in my one part of my filter is out of my image. So I need some padding in order to avoid this one. So I need some padding. So in the case, the input volume, I will pad in all directions with zeros in order to avoid such a disruptions in the image during the process. And then this padding helps us to control the spatial size of the output volume. So whenever I don't use any padding, then the feature vectors that have been generated or the feature maps that have been generated will not be in the same size as that of my input image. So in order to avoid this one, I'll go for the padding also. So I'll try to pad it in all directions, that is both from my top, left, right, and all directions. I'll pad it with the 0 values here. So these are the 3 parameters which needs to be considered whenever you move on to the convolution layers. So coming to the summary, so I just have an image of width W1 into H1, and the depth of the image is D1. So we discussed about 4 different hyperparameters. It is the number of the filters. So as I told you, it is this depth, number of the filters decides on the depth of the volume layer, volume input layer. So this number of the filters, and then what is the size of each and every filter that have been used, whether it is a 3x3 filter I am applying or a 5x5 filter I am applying. So it is also one such hyperparameter that will be used. And then the strides, how about the stride? Is the stride is 1 or the stride is 2? And then the amount of 0 padding, whether I am going to pad something, some values, whether I'm going to pad 0 values onto my image or not, I am going to pad the values. So all these are considered to be the hyperparameters. So based upon this one, what will be the size of the image that will be created, or size of the feature map that have been generated from this convolution layer. So this W1, H1, D1, based upon these 4 hyperparameters, will produce as an image, produces a activation feature map of size W2 into H2 into D2, where my W2 is nothing but W1 minus the number of filters plus 2 into what is the padding amount by S, the number, the stride. If the stride is 1 or stride is 2, S is equal to 1 or S is equal to 2 plus 1. Similarly, my H2 will be H1 minus F plus 2P by S plus 1. And then my depth is nothing but the number of filters. So this is what. So after applying my convolution layer, based on the number of filters I have used, what should be my size of my feature map? What should be the size of my feature map is nothing but W2 into H2 into D2. So for example, if my input image is of size of 5x5, and then the depth is 3, that is 3 channel image, then the convolution parameters will be 3. That is where I have used my two filters I have used, which is of size 3x3, and I have used a strided up S is equal to 2, and then P padding is equal to 1. So this is what it will generate an image of size 3x3, feature maps of size 3x3. So this is what is this summary. So the purpose of the convolution layer to extract the features. Once these features have been extracted, once I obtain the dot product, the summation of the dot product, it has to be given to the activation functions. So the purpose of the activation function is to decide on which part of the neurons has to be fired and which has to be passed on to the next layer. So I can also say the purpose of this activation function is to introduce a non-linearity into a system, that because in order to that basically because all my operations we have discussed, my convolution operation is a linear operations. So in order to introduce a non-linearity in functions, I am using an activation functions. So most probably the activation function that will be used in the earlier layers will be a ReLU activation function. What the purpose of the function is to suppress the negative values. So I can say f of x is equal to max of 0, x. So it is maximum of these two values will be obtained. So this is what is the ReLU. So this layers just changes all the negative activations to 0. So I don't have any negative activations after this activation, after applying this activation function. So the next layer, which is what is said to be max pooling layers. So I have generated an activation feature maps, and this feature maps is considered to be of some dimensions. So with the help of this feature maps, the number of parameters and the computations in the network will keeps on increasing. So the purpose of the pooling layers is to reduce the spatial size of the representation. So the feature maps are nothing but the representations of the images. So I need to reduce the spatial size of the representations. Because of this, whenever the number of parameters increases or the computations in the network increases, it leads to overfitting. So in order to avoid this problem, we are introducing a layer which is what is called to be a pooling layer in between the convolution layers. So after each convolution layer, there will be a pooling layer. There are three different types of pooling layer. One is the max pooling layer, min pooling layer, and average pooling layer. So whenever it is a max pooling layer, whatever the output from the convolution layer, it will be divided, the size will be reduced by 2. So what is the purpose of the max pooling layer? It will take according to the size of my pooling layer, that is according to the filter size of my pooling layer. For example, if it is 3x3 filter pooling layer I am, or 2x2 filter size I am using max pooling layer filter size, then it will take each 2x2, find out the maximum value in the 2x2, that will be taken as my output. Similarly, it will consider every 2x2 part of my feature map, and then I will try to reduce the spatial size by taking only the maximum value present in the 2x2 region. So by taking the maximum value, meaning that it will takes the high level features, or it will take the features which is highly contributed in the images. So this is what is this one. But in case of this one, the depth of dimensions remains unchanged here. But the spatial size of the representation of the feature vectors have been reduced in some form. But in case of my minimum pooling layers, the minimum value between the prescribed size of my filter will be taken into consideration. In case of my average pooling layer, if the filter size is 2x2, the value have been averaged, and that value will be taken as an output of this part. So this is what. So I have a single depth slice. This is what is an feature map. So whenever I am saying it is a 2x2 max pooling layer, I am applying. So I will obtain this one. This one is the maximum value that has been taken into consideration, and this one is the maximum value taken into consideration. This one is another maximum value taken into consideration, and this one is the next maximum value taken into consideration. So my 4x4 feature map have been reduced to a size of 2x2. Yes, here I have used a stride of value 2. So the next layer, which is what is called to be a dropout layer. In case of this dropout layer, what is the purpose of dropout layer? It is not always necessary that all the neurons is just to be propagated from one layer to another layer. So we can randomly drop some of the neurons with the help of the dropout layer. But which one? This also will suppress the problem of the overfitting. It handles the problem of the overfitting. But the number of neurons to be, we can specify in the dropout layer how many percentage of the neurons needs to be dropped. But these number of neurons is randomly picked at each time of my epoch, and it will be dropped out during the training. And this cannot be used during the testing time. This cannot be implemented during the testing time. And this is what. So I have extracted the features. Once I have extracted the features, I have reduced the spatial dimension of the features with the help of the max pooling layers. Once it has been done, it has been given to a dropout layer. If this dropout layer is an option one, if needed, you can include it in your network. If not, you can leave it off. So then I need to flatten the feature vector. So I have the feature maps here. Now till now my feature maps is in the 2-dimensional format. Now I need to provide it to my classifier. My classifier is a fully connected network. So I try to flatten the inputs. I try to flatten my I try to flatten the things with the help of my flatten layer. So this pooled feature map will be now flattened into one column. As one column. This is what is the map that will be generated after introducing my flatten layer. So this can be till now I have used a classifier. Till now I have used the used as my layers as a feature extractor. Now, till now I have extracted the features. The feature maps now been flattened and it is in the form of one column. Now it has been now be given to a classifier. The classifier is a fully connected layers. So we all know what is this classifier is all about. It is a fully connected layer. So where each neuron of the this whenever it is a classifier, this like from here each of these neurons will be connected to each other neurons present in the next layer. So this will be classifier here. So this is otherwise called to be a dense neural network, where each of the neurons from each layer will be connected to each of the neurons in the next layer. So the output of the classifier, it all based upon whether it is a binary classifier or a multi-class classifier. If it is a binary classifier, my activation function at my output layer will be a sigmoid activation function, where it will give me a probability value of my features. Or if it is a multi-class classification, I should have a categorical I should have a different activation function in the output layer. So this fully connected layers where each neurons of every layer will be connected to each other neurons in the next layer. So as I told you, at this output layer we make use of a cast function in order to find out the cast value, or in order to find out the errors. This error can be obtained by finding out the difference between my predicted value and the ground truth value. So this can be calculated with the help of the cost function, which is what is called to be a loss function in the case of my layers. And these loss function can be a categorical cross entropy or a binary cross entropy, depending upon what type of classification I am undergoing. If my classification is a multi-class classification, I can make use of a categorical class entropy, or if it is a binary classification, I can make use of binary class entropy. So I can compute the error in the prediction, I can back propagate the error, so that I can update my layers will update my weights and bias in order to reduce the errors. So I keep on using this methodology in order to train my images, in order to extract the features, and in order to train my model such that my model will find the discriminating features from the input images that has been given in order to perform the classification. So this is what is all about your convolution neural network. I will move on to the case study. Once we have done with that one, then we'll move on to the transfer learning technique, and I'll explain about the efficient at present in the transfer learning technique. I'll give you a link now. You can use the link where you can find the data set, everything. You can download it and put it in your drive, in your Google Drive, so that you can work along with me. I have pasted the link in the chat box. Can I wait for you or what?
Ma'am, I'll give them 5 minutes and then.
Don't do anything in the drive itself. You download the drive and upload it in your download the folder and upload it in your Google Drive.
Ma'am, excuse me, ma'am.
Tell me, ma'am.
It's asking permission, ma'am. Unable to open.
One minute. You are not able to open the link itself, ma'am.
Yes, ma'am.
Okay, wait, ma'am. I get you another link.
I think it might open now.
Oh, yes, I'll again post the link. Now, can you use this link and check out? Tell me, ma'am. Is it possible for you to open it now? Yes. So now what you need to do, don't do anything inside this one. Download the link and upload it in your drive. I request everyone to download it and upload it, because if you do some changes, it will affect me also.
Ma'am, whether you have shared something, ma'am?
What I have shared the link where I have kept my code and my images. You can download that one and then you can upload it in your drive. I request everyone, don't open it and do something in your in this thing. If you do that one, it will affect mine also. So kindly download it from the link and upload it in your drive. You go to your Google Drive, give new folder, then upload this entire folder. Can anyone able to do this one? Can you hear me?
Yes, ma'am.
Everyone is able to do this one?
Yes.
Kindly
Yes, ma'am.
answer.
Yes, ma'am. Yes, ma'am. Yes, ma'am.
Yes. So it's a simple neural network that I have used here. Now what images I have used, this image, what I have used is liaison images, skin liaison images. So where I have obtained this image, it is an image that I have obtained it by performing a task. It is ICCIDS. It is International Skin Imaging Collaboration. This is the task. This is one forum where it has been conducting some tasks on skin images only, skin cancer images, and this forum has been started at 2016. From 2016 onwards, they will be releasing some set of images and they will be conducting some tasks like classification, First, they have started with the binary classification, and they have released some images on multi-class classifications, and they asked us to do the task of segmentation and do the classifications. So each and every year, the forum people keeps updating the number of images and keeps introducing complexity in the images and introducing new tasks each and every year. Now this task is also open. The task 2020 IASIC 2020 is still live now. What you can do is you can go to the link, download images, you can apply any models or anything. You can, after obtaining your results, you can upload your results so that they can check your accuracy and they can give your accuracy of your results. So this is what for the test images, they can check it with your model and then they can provide your accuracy of the results. So this is what this images that have been taken from the ISIC 2019 task, where it is a binary classification task. So we know cancer is of two types. Basically, it is of two types, melanoma and non-melanoma, or we can call it as a benign or melanoma. Benign may or may not be a cancer, but melanoma or cancer cells. So I will show you one type of image first. I'll show you the sample images. I think somebody is running the code by opening it. If done that one, please don't do this one. If you do some changes, it will reflect in my thing. I kindly request everyone to click onto the link, download the folder and upload it in your drive and work on your own.
But Nalini ma'am, to avoid this, now everybody have downloaded it. Now you can
Yes.
remove the editor option.
I don't think that I can remove it. I will check it out.
You can able to ma'am unshare it. Okay, you have given the link. We have not shared it. Restrict it, ma'am. No, go to the share option. Keela, anyone can internet with this link, can we, change it? Change that anyone with the link. Commenter. Share. Down, viewer. Commenter. No, anyone anga click pannunga. First, anyone with the link you have arrow mark, no? First, on the left side. Change that. Restricted. Yes.
Done.
No, it will be changed. It's restricted.
Okay. I will show you a sample melanoma and non-melanoma images. So I'll show you at the end which is melanoma and which is non-melanoma, but it is what I have taken is for the binary classification. So as we have discussed, I am using a Keras network model here, a Keras platform here. So it will undergo all the steps that we have discussed. The first one is I need to create the model or define the network. And then once I have defined the network, then I need to compile the network. and I have to evaluate the network by providing the value, and then I can predict it from the one. This have been done, my model will be created, and then I will have to predict the test data using the model. This is what way I'm going to do. So for this one, I need libraries and functions. This, as I told you, the TensorFlow is on the top of the Keras. So I am just importing the TensorFlow, and then in order to build the layers, the basic way of building the layers is what is sequential building. So I will define each and every layer sequentially in sequence. So I am just importing sequential. If not, if it is a complex, we have another option, which is what is said to be functional. So I am not going to deal with that part. I am going to use a sequential layer definition. So I am just importing sequential. Apart from it, I am going to use an dense function, dense layer, activation layer, and then flatten layer. And then I am using a max pooling layer, and then I'll be using a dropout and dense. So this is not needed. I have already defined the dense, so I can remove this one. So these are the layers I'm going to define. And then I have to import all my activation functions. So I am just importing from the Keras all the activation functions. And then this utilities, all these OS and all these paths and everything is needed for me. OS, utilities and pill images, everything is needed for me in order to open the corresponding path where I have stored my images. And then to read the images from this one and read each and every images, I need a pill.image library also. And then this npy, as I told you, each image will be considered as an array, as a matrix. So it is a numpy array it will be considering. So I need to import this numpy array too. And then this MATLAB plot, the end I have plotted the validation and training accuracy, and two validation and training glass. So I need, in order to plot it as a graph, I need MATLAB lib. So I am downloading all the libraries and functions. We are importing all the libraries and functions, which helps me for building the CNN model. And then I am just mounting it. I am just mounting it on my drive. So all these have been mounted in my own drive. So I am just giving from google.colab import drive.mount. Yes. And then, as I told you, the first step is to define the network. So I am just here in defining the network. It is a sequential way of defining the network. So I am just saying model is equal to models.sequential. The first layer I am introducing here is the convolution layer, where this convolution layer consists of all these parameters. One is the filters, number of filters I am using in order to do the convolution operations. The next one is the kernel size. It is nothing but the size of the filter. And then the third one is the stride. How about my stride? By default, the stride is 1x1. That is, I will stride each and every pixel on my image. And then the padding. The padding, as I told you, it will helps me to retain my size of my feature vector similar to that of my, equal to that of my input image. So there are two different types of padding. One is valid, the other one is same. So whenever I am saying valid, I will not be using any padding technique there. But whenever I am saying same, I will be padding my outer layers with in all directions with 0. And then these are the four basic things that we usually use. Apart from it, we also say what is the activation functions, and also I'll say what is my input shape. This is what is the input shape of my image. So I am just taking an input image of size 224 cross 224, and then it is a 3-channel images. So I am just saying 3. Yes. So the number of filters that have been used is 32, and then each filter is of size 3 cross 3. Yes. So the activation function, as I told you, I am using a ReLU activation function. So it will suppress all my negative values and will have only the positive values that will be passed on from this layer to the next layer. And then this is all about, and I have not defined about the stride and padding here. So it will take the default stride of 1x1. So I will stride each and every pixel of my image, and my padding value is all about valid. So I am not going to do any 0 padding here. And then once it has been done, the convolution operation have been performed on my input images. It will generate a feature map. As I told you, what will be the size of my feature map will be W2 into H2 into D2, where W2 has to be derived from all my 4 hyperparameters and also this one. So this is what I obtained. This is what will be the size of my feature map. Yes, and then this is what the number of filters I have used in order to obtain this feature map, number of filters that I have used in order to obtain the feature maps. And then this has been passed on to I'm defining my next layer, which is what is said to be max pooling layer. So as I told you, the purpose of the max pooling layer is to reduce the image size by 2. So earlier my size at my convolution layer is 222 cross 222. After applying the max pooling layer, it has been reduced to half. It is 111 into 111. And again, it is also using 32 filters of size 2 cross 2. And then I am adding, and so I am adding one convolution layer followed by a max pooling layer. Similarly, I am adding 3 sets of convolution layers and 2 max pooling layers. And then so at the end, so when you look on to this each and every convolution layer, my first convolution layer has a filter of 32 filters, and in case of my second convolution layers, I am just introducing additional filters, that is 64 filters have been used. And in my last convolution layer, I am using the same filters, that is the 64 filters. Yes, so at the end of this convolution layer, I get a feature map. This is what the feature map. So this 52 cross 52 feature maps have been obtained at the end of this convolution layers. Now it has been given to my flattened layer, where I am trying to flatten my feature maps into a single vector. So this is single column. Once it has been flattened, till this part, I can say it is a feature extraction part. I have extracted the features, done the convolution operations, and applied to the max pooling in order to reduce my spatial resolution. And then similarly, I have 3 sets of convolution layers and 2 max pooling layers. Once it has been done, it has been given to a flattened layer, and then these feature vectors now being given to the dense layers, which is called to be nothing but the fully connected layers. So the first dense layer has 64 filters, and my second dense layer, which is what is called to be my output layer, where I have a dense, why I am giving it as a 1, because it is a binary classification. So based on the probability value, I will decide whether it belongs to my class 1 or class 0. I'll consider my class 1 as a melanoma and class 0 as a non-melanoma. Totally, in my training images, I have in this 2019 ICICIDS data set, there are about 25,000 images. The problem that is present in the data set is all about the class imbalance problem. That is, the melanoma is the melanoma, the total number of melanomas present in the data set is very less when compared to the number of non-melanomas present in the data set. So in order to implicate that one, I also taken a data set, which is a very small range of data set of about 281 images. And of these 281 images, I have taken 81 as my melanoma and 200 as my non-melanoma. So in order to implicate the class imbalance problem here also. So as I told you, at my last layer, I should have I use an activation function, which is what is called to be a sigmoid activation function. Now I will run this model. Yes, this is what is said to be my model summary. It gives me the summary. So what are the layers? What is my input shape? And what is the output of this one? How many parameters have been generated at each and every point? This is what is my model summary. So I have been generated using my network. Total parameters is this one. Of this much of trainable parameters. All these parameters have been taken into training, because I don't have any dropout here. So none of the parameters have been dropped out here. So all these will be taken into consideration here. And then, so this is how about the definition construction of a network. I have constructed my network. Now I need to move on to my data set. So as I showed you, there is 2 things. One is the folder, where the folder is consists of some images. As I told you, there are about 281 images. Of this, 81 belongs to melanoma class and 200 belongs to non-melanoma class. And I have a file, which is what is said to be train.csv file. This train.csv file contains metadata about the image. That is, it talks about what is the image name, what is, I don't want the patient information. I am not going to use any of the patient information. And then patient ID. So, and then I have the sex, age approximations, and then all these some meta informations about the patient and also the information about the image is also be present. So it is says whether it is a benign and malignant, it is benign and it has a target value of 0. So this such of information, what is the width and height of the image, and all these informations have been present in this metadata. So what I will be trying to do is here, when you look onto this one, normally in case of any supervised learning, I will be using, in case of your deep learning model, I'll be directly reading from the folders. So I have the number of classes is nothing but number of folders present in my training set. But here I don't have this such a type here. I have put all these training images in one folder, which is train, and then I have to read it from the CSV file, which says which images, which class. So this will be taken as the labels will be taken from the CSV file. This is how I have coded in my content. So what I will be trying to do is I am going to my drive, and I have a folder called main folder called ICCIDS 2019. Inside this one, I am having a training file. So star, which represents all the images, has to be read. So I will read all these images. I will resize the image to a size of 224, and then I will be converting into an NP array, and this will be stored in my X underscore string. So all my training images have been moved on the variable, and it will be stored in the form of an array in this X underscore train. And I want to confirm what is the size of this one. So there are about 280 images, and then the shape says what is the number of images, what is the size, width and height of each images, how about the channel of the images. So there are about 3 channels through RGB channels have been present here. And then I am trying to read the CSV file. I am reading my CSV file. For that one, I am using PD. PD is nothing but the panda. With the help of the panda, I am reading my CSV file, which is again present in my ICCIDS 2019 folder train.csv. And then I am training this one. Y-train will get all this information that has been read from the CSV file. Of this one, my target says the labels of my images. So I need to extract the 7th column. So I am saying ytrain.I location for every 1 to all the images. It is starting from the first row to the end of the row, that is 280 row. I am just reading out my 7th column, which is nothing but the labels. So my ytrain will now contains the image number and also the corresponding labels for the numbers. So 1 and 2 represents it is a non-melanoma, 3rd represents it is a melanoma. So this is what is the, so I am just getting ready with my data. I have fixed on my training data, and also I have correlated my training data with my labels. Now I have ytrain, where my ytrain will contains the training data informations and the corresponding labels in it. And then for the convenience of the computation, I am just converting it into float. So I am just converting it the type into float, and then I am putting all these class labels in the ytrain. So my xtrain will contain the image and ytrain will contain the class labels in the float format. And then now I have something called train test split. So I don't have a separate folder for training and separate folder for testing. I have a single folder, which is what is said to be the training folder. All my images, either it belongs to a class 1 or class 0, all these are present inside the single folder. Now I need to split it. What should be my training images and what to be my testing images. So I am splitting it. Of this, all the images of about 280 images, 0.33%, 33% of my images has to be considered for testing, and 70% of my images has to be considered for training. So my ytrain is the image for the training, and xtrain will be the images of my training, and ytrain will be the corresponding labels of the training images. And similarly, x validation, that is nothing but test set. I am just considering my test set as my validation set. X validation is the validation test set images, and y validation is the labels for the test set images. This is what we need to predict it, but in general it has been given for ground truth. For comparison, I am just having this y validation value too. So now I have now my data set is also I am ready with my data set. Now I have created my layers. Now I am ready with my data set. Now what I need to do is I need to compile my layers. So in this case, I am using an optimizer and a loss. This is the loss functions, which helps me to I evaluate the or find out the differences between the predicted value and the targeted value. Here I am using a binary classification, so I am using a binary cross entropy. My optimizer, my job of my optimizer is to reduce the learning rate. So it's such that within a some split time of time, my model has to be learn everything. For a split epochs, it has to be learn everything and should be get ready the model. So that is what is the purpose of my optimizer. So I am using an Adam optimizer, and for the purpose of the loss evaluation, I am using a loss function, which is what is called to be binary cross entropy. And then the metrics used to evaluate my system is what is called to be accuracy. So I am going to find out my accuracy. So accuracy is what is my input, what is the predicted value, and what is my ground truth value. Comparing these two things, I can get what is my accuracy. So after this one, after compiling my model with my loss function and my optimizer, I am just fitting my model onto my data. So X train and Y train is my model, and I am just training my epochs, 5 epochs. I am just going 5 epochs. Since I have normally a deep learning model will consider a larger amount of data, but here since we are using some small amount of data, considering only a small amount of data, we will be thinking that each image will be taken and it will be trained. It is not so. Whenever the image is very larger, it will be divided into some batch size based upon the total amount of images and the number of epochs that have been taken into consideration. It will be divided into some batches. So each batch will have some set of images. So this training will be done under each epochs for each and every batch. So this batch size will randomly changes for the next epoch. So this is how this one, but here I didn't define my batch size. It is default. It will take by default, that is it will take the total number of images, divides the number of epochs, so that it will consider the number of batch size, number of images in each batch size. Here all my 280 images have been divided into 6 batch sizes. So this is what I am doing. I am running it for 5 epochs. So I am just putting the value into the model and I'm running it for the 5 epochs, and this is my validation data and this is my training data. And then it undergoes each and every steps. It consider each and every batch of the images. Every image in each batch will be taken into training, and then it evaluate the loss. And it is what is this one is what is said to be the training accuracy of the images. And based upon this epoch, it is just validating my test set, that is xval, and it will be finding out the accuracy based on the predicted value and the y value. So my validation accuracy is 0.318 at my first epoch. Similarly, I am undergoing a 5 epochs. At last, I achieved an accuracy of validation accuracy of 0.59 and a training accuracy of 0.738. So I am just displaying this one. What is my accuracy after all my 5 epochs for all the batches? So it is all about my loss is 6.760. So it is the average of this one, and my accuracy is all about average of this one. It is 0.7326. And then in case of my testing, my validation set, this is what my loss is all about this one, and my accuracy is this one. As I told you, my hyperparameters works out here. What I need to do is, this is a model, this is a deep learning model, so I need to use a large amount of image. That's what is the first thing I need to do. Apart from it, I also can increase the number of epochs. Yes, I can also include my strides, and also I can also increase my padding technique also, so that I can get more accuracy in case of my large amount of image set here. And then as we know that I have used my accuracy as my metric, I'm just getting it what is my accuracy. So I have something called history of accuracy. I'm storing it in the variable accuracy, and validation accuracy also I am just getting it, and then using the inbuilt function that is available, and I'm also taking what is the loss of my training loss and the validation loss, and using this one I am trying to plot a graph where my graph is between the accuracy, training accuracy, and the testing accuracy, and also training loss and the testing loss. So I have two things. One is the blue color line and also the orange color line. This blue color line is the training accuracy and the orange color line is the testing accuracy. So my training accuracy keeps increasing. At one point it comes down and again it keeps increases. But in case of my validation accuracy, it increases and it has been in a straight line for larger amount of time and then it keeps decreasing here. So this is what is between the accuracy. So in my y-axis, in my x-axis, it is the number of epochs, and this the x-axis, y-axis, it is what is the accuracy values, training accuracy values and the testing accuracy values. Similarly, a graph has been plotted for the loss also. It is the training loss and validation loss. So it is all between the x-axis and y-axis. It is all about the epoch and the training and validation accuracy. So this part of my code helps me to plot my training and validation accuracy. So now I need to do the prediction. So I am just predicting the model. So I am just giving the validation of my model. How I am predicting is, as I given a sigmoid function, it will give the probability value. So based on the probability value, I need to say whether which class the predicted image belongs to. So if my predicted value, I am just fixing a threshold, is greater than or equal to 0.5, I can say it is class 1. If it is less than 0.5, I am just saying it is a class 0. This is what is said to be the predicted value. So this is what is the probability value of my y predict. So this has been getting displayed here, and I hope that everyone knows about the confusion matrix. So what is a confusion matrix? What is the use of a confusion matrix? Can anyone tell me what is a confusion matrix? Why we are using a confusion matrix?
To evaluate different measures based on positive rates and negative rates.
Yes. So in order to evaluate the performance of my model, I am using this confusion matrix. So it all depends upon all these 4 values, that is true positive values, false positive values, false negative and true negative values. So this I am just providing a confusion matrix between yval and ypred. And then this I have a inbuilt function, which is what is said to be the classification report, which says about what is the precision value during my training for my class 1, and a precision for my class 2, a recall for my class 1 and class 2. My F1 score is nothing but precision by recall. So that is what is this this is all about. And again, I achieved an accuracy of about 0.59 in case of my test case. So this has been given printed here. So this is all about this classification report too. So this again, I'm telling this is a simple model where I am just introducing you how a model can be built using a Keras platform. So you can add, similarly, you can add n number of models. If you want to add a batch normalization, you can add batch normalization. If you want to change the activation function, you can change the activation function. If you want to make a deeper network, you can add more convolution layers, and instead of a max pooling layer, you can also try it out for a different layers. So this is how you can change the network, and you can check it out how it is working for your particular input data. So this is all about this convolution neural network. Is there any doubt in the part of the code?
No, ma'am.
We'll move on to the next part. This is all about the transfer learning technique. Yes. This what we have done is we simply created using our different layers. We built a network and we will try to build a model using this network. And then you have something called a transfer learning technique. So whenever we try to train a deep neural network from scratch, it will take a lot of time, and it needs requires large amount of data, and also it requires large amount of resources like GPU and the all these is being required in order to train and tune a neural network from scratch. So the cheapest way and the fastest way of adapting any deep neural network is by exploiting the generalization properties. So I look onto the generalization properties of the image. If one particular model is providing good results for a particular data set, if I am having another new data set, I will check out with the generalization properties of my data set. If both are considered to be very similar, I may make use of the model that have been used for the earlier data set. For example, we have as I have discussed, there are in the international level, there are large number of tasks have been conducted for an image. One such task is what is said to be image. One such data set, large number of tasks for computer vision have been conducted each and every year. So once for one large data set which being used under the computer vision task is what is called to be ImageNet data set. There are about some millions of data have been present in the ImageNet data set. All these elements are considered to be millions of data sets are there. All these elements are considered to be the millions of image data sets, and all these elements are considered to be the general images. So large companies have worked on these images and they have built their own model in order to define they have built in the old model in order to perform some computer vision task. So these models informations are this model have been so instead of we trying to create a model which utilizes a large time and a large resources, we try to adopt these models for our new images and try to get some results for this one. This is what is said to be transfer learning technique. So transfer learning is nothing but it is about leveraging the feature representations. Pre-trained model is what is called to be a transfer learning here. So this transfer learning technique will be using a massive data sets. This pre-trained models will be using a massive data sets. We know that, as I have discussed, large number of tasks have been conducted, large amount of complex images will be created in this task for each and every task, and great companies, larger companies will be working on this task, and they are providing some models on it. Which produces a good results for the particular data sets. So those models are called to be if I am using those models for my new data, I am just leveraging some of the properties of this model and will be using this one, some of the representation of my models, because each models will try to extract the features, and from the features it will try to classify it. So all these images, whatever be the images, either it is a medical image or it might be normal images or general images, the basic features are the basic primitives like lines, edges, colors or something like that. So that's why I told you the lower level features will be the basic primitives. The higher level features will represent the higher level features will give you the the higher level features will give you the discriminating features of the particular image set. So this pre-trained model whichever being used on the massive data sets will be used for our purpose also, for our own data sets also. So I have reused one such model, which is what is called to be EfficientNet. So this in case of any transfer learning technique, we will follow 6 steps. The first step is we need to first obtain the pre-trained model. Once this model has been obtained, we need to take the database, that is a base model. That's as I told you, the base model is nothing but the lower level features, where these features helps us to extract the low level features, and then I can freeze the layers. That is, as I told you, I can freeze all my lower level layers. Freezing means I don't want to train my pre-trained model with my new data. So all whatever the models I am talking is maybe a deep neural network which consists of some hundreds of layers that has been present in it, which is possible to handle a complex data sets too. So I will try to freeze some lower level layers. Keeping it in mind, all my lower level features will extract the basic primitives from my images. So I'll freeze the lower level layers and I will add some layers at my top depending upon what is my requirement. For example, if one particular model, if it is my AlexNet, the image data set, it has been trained for about some thousands of classes. For example, if I have been taken that it has been trained for thousands of classes, what now I am using in my data set, it is of a binary classifications. So yeah, in my output layer of my AlexNet, that will be 1,000 neurons that will be present there. But in case of my data set that I have been taken, it is not so. So what I will be doing is I will be removing the top layers, and then I can create my own layers, which is necessary for me. And then I will try to train, I will take all the weights, all the features, all the weights from up till my freezed layers. And then I will train the model with the newer layers with my new data. So that is train the new layers on my new data set. And then if I want to improve the model, I have to do the fine tuning. That is, I can improve my model. Either I can unfreeze some of my lower layers, some other layers, and I can train it with my data so that I can do some fine tuning in this part. So in case of my transfer learning steps, there are two type of steps that can be used, where my transfer learning technique can be used as a feature extractor alone, or my transfer learning technique can be used as a fine tuning. In case of my feature extractor, I will extract the features directly and give it to any classifier. Either it may be a SVM or something like that. I can extract the features, give the features to the classifier and try to classify it. Or otherwise, I can I can try to unfreeze some of my layers, try to train the model in the layers, and then I can classify this one. So this is what been done in case of the transfer learning technique. So I have used one such model, which is what is said to be the efficient net. So what is advantage of having the efficient net? Till now I have talked about some hyperparameters. So this hyperparameter tuning will make my model as a good model, or I can say it will provide me a better accuracy. For all these hyperparameters tuning, I need to first check it out. What are the hyperparameters available? How about my data set? Which hyperparameters will affect my data set, or which parameters will affect my accuracy? So I need to know all these in details, and by trial and error method only I can set up on my hyperparameter things. But in case of my efficient net, it is not so, where it has been proved that doing some hyper tuning, it is the accuracy of the model will keeps increasing there. So by balancing scaling of individual dimensions, that is either the width, depth, and the image resolution of the input, will keep increasing the accuracy. So taking all these into consideration, this efficient net make use of one technology, which is what is said to be compound scaling method. What it will does is it tries to balance all the dimensions of the network, that is in the width of the network, depth of the network, and image resolution of the networks. All the dimensions of the network will be taken into considerations, and it will try to balance it against the available resources. So that will improve the overall performance of the thing. So this is what is all about the thing. So this balancing this dimensions of the network, this being happened in the efficient net with the help of a methodology, which is what is said to be compound scaling method. How it does it? It does a grid search. How, so what is the relationship that exists between the different scaling dimensions and the base network? There are about different variant of efficient net, starting from B0 to B7. Each of these efficient net are different dimensions networks, where it have been where this balancing can be done with the help of the grid search, in order to find based on the available resource constraint that has been present there. So this constant resource constraint has been fixed, and based on the resource constraint, how should be my width, what should be my depth, and what should be my image resolution, all these hyperparameters will be decided internally by the efficient net itself. So whenever I am talking about the width, the number of filters, how many filters have to be used. Whenever I am talking about the depth, number of layers, how many layers should be there. And whenever I am talking about the image resolutions, any image of any input images of complex resolutions will also taken into consideration. So all these will be balanced based upon the compound scaling method, and it will try to, it has been fixed based on the fixed resource constraint. And then why this have been used is whenever this efficient net have been used on my ImageNet data set. As I told you, this is a large data set where it contains of about 1800 classes or something like that, and then there are about 1 million images present in the class present in the ImageNet data set. So this ImageNet data test, when we tested this ImageNet data set, this MobileNet and ResNet, based upon this property, that is compound scaling method, have given a better results. There will be increases of about 1.4% in accuracy with the help of the MobileNet, and above about 0.7% of accuracy in the help of the ResNet with the help of the compound scaling method. So these two things have been considering as the layers of my efficient net. So this is what is my efficient net. It is what is all these reddish color can be called to be a mobile inverted bottleneck. What is this? Each mobile inverted bottleneck consists of some residual blocks, and there is some skip connections that exist between the residual blocks. So this is what is the importance of this mobile network box. So I have a convolution layers. After this one, I have some set of mobile net convolution layer, and then for this is what is said to be my layers of the efficient net. So what I am doing is I am going to use this one. I am going to use this efficient net in my transfer learning approach, and I have done a small work on this one. So this is similar what we have discussed till now for your convolution neural network. So I have introduced all this, imported all my low libraries and functions needed for this one, and then here I have done one important thing, one thing apart from which have not been done in my CNN. That is what is said to be sequential network. So I request, I got some information that you people are doing directly in the drive. So I request everyone to download the content and do the things, because the drive have been shared with n number of people. If you do some changes, my code will get changed. Yes. So this is what is augmentation technique. So this augmentation technique helps us to generate, or to not to generate the images, to produce some images which will be useful in training. That is how this generation have been happening. I am applying some transformation technique onto my input image, training input images, that is random rotation. So I am just trying to randomly rotate my images so that I get some other pair of images, because information present in the images is not going to change, so that whenever I rotate the images, so random rotation, random translation, random flip, and random contrast. So all these are this information that is present in the all these transformation technique. I am using this one in order to generate some more images that will be helpful for doing the training. But I can say what type of transformations can be used, but I cannot. This is again a black box. I don't know how many images have been generated with the help of this transformation. So this is all of my image augmentation. So I have done my image augmentation in order to produce to add some more amount of images onto my training set. And then this part is what is said to be MobileNet V2 pre-process input. This is Keras has some inbuilt pre-processing technique used, and this will helps us to apply the basic pre-processing technique that can be applied onto my image. So I am just reading all my images, I am taking my images, I am applying some pre-processing, and I am just putting it in the pre-process input. And then I have defined a class here. The class is to build the model. So I am just passing the number of classes. Yes, ma'am.
Ma'am, you didn't share the screen, ma'am. Coding part.
Sorry. Can you able to see it now, ma'am?
Yes, ma'am. Yes, ma'am.
Thank you, ma'am. Yes. So this is the sequential part where the augmentation technique, where I am applying some transformation to my training images itself. And then I am trying to generate some more additional images onto my training images. And then I have defined what should be my size and width of my image. And then this part is nothing but a pre-processing step. So basic pre-processing step have been available in the Keras as an inbuilt. So I am just making use of this one in order to apply basic pre-processing technique onto my images. Now I have a model. I have defined a class for building the model. I am just passing the number of classes here. And then, so just it will take what is my input shape and size, and it is going to take a 3 channel images here. And then this, as I told you, I am applying some augmentation technique. My inputs have been passed on to the augmentation technique, and the augmented images have been stored in X now. So now X will contains all my images. Now I am just calling my efficient net model. That is the basic model. It is the B0 model. There are different variant of efficient net from B0 to B7. I am just using only the B0 model here. Now it is a transfer learning technique. As I told you, it is possible for me to freeze any of my layers, and then I can unfreeze I can train with my new data to the unfreezed layers. Either I can do that one, or I can remove my layers and add new layers so that I can train my new data with this layers here. So include top is equal to false. I am not doing any training here. I am just getting my weights. These weights are nothing but the weights that have been obtained by training the model using the ImageNet data. So I am just getting all my weights of my ImageNet model, and then this X is nothing my input images that have been passed here. Yes, I am my top layers have not been trained here, and then model trainable is freeze. That is, I am freeze the pre-trained weights. I am not going to do any training here. I'm just freezing all the layers of my model, and then this one I am just rebuilding my top layers. That is, I am removing the classifier part. I am rebuilding the top layers with 2 layers. One is I am just adding global average pooling layer 2D, and then I have a batch normalization of X. So this X will contain my input data. It has been normalized into batches, and it has been passed on to the global average pooling layer. And then I have used a dropout layer of about 0.2%. So 0.2% of my neurons will be dropped out at this stage. And then I have added a dense layer, where my dense layer will act as a classifier. Try to classify it based on the number of classes. It is a binary class, and I have a sigmoid function of this part. And then I am just passing the input data onto this, process the input data onto this one. So this will be acting. So I am just removing the top layers, which has been already present in my efficient network, adding a layer of average pooling layer, and then at my end I am adding a dense layer which acts as a classifier now. And then I am doing the compilation. This is the model compile. I'm using an optimizer. This optimizer is an Adam optimizer. I am also specifying my learning rate, that is LE-E. And then as we have discussed earlier in my CNN, I have used a binary cross entropy as my loss function, but here I am using mean square logarithmic algorithm as my loss function here. Yes, I am trying to compile this one. This compilation where I am using what is the optimizer I have defined. I am using an Adam optimizer. This will be considered. I'm using an mean square logical error. This will be considered. I am using a metrics to evaluate the system is accuracy metric. Yes, so this function will return me the model after compiling the model. And then I have defined the number of classes. It's a 2 class thing. I am just building the model. I am just calling the function, and a model will be written back. And then I have defined what is my number of epochs, and I have also stated the summary. I want to display the summary, so I have stated the summary. You just look here, all the layers of my efficient net. So this part talks something about my augmentation technique. And these are the different blocks that is available. Even though I am not defining your each and every layer of this one, it is some residual blocks, some set of blocks present there, and you have some multiple number of residual blocks that is present there, and it will make use of skip connections. That is normally you will pass the neurons from previous layer to the next layer, and from the current layer to the next layer. But here in case of a skip connections, the layers, the informations will be passed from n to n plus 1, and similarly n to n plus 2. So similarly, this is how this information keeps passing there. So more the informations, more number of features will be extracted, and this will be helpful for getting more accuracy here. So this is what is the blocks of your efficient net. And the same thing that I have repeated, I have taking my data from the same folder, train folder, and I am reading my label from my train.csv, and then I am just splitting the data set, that is train split, as a 0.33 as my testing and training, and then I am just fitting the model. So fitting the model.fit, giving my input, number of epochs, and then validation set. So it undergoes the number of epochs, and the model have been evaluated, and it found since the model is a complex model, the number of images used in my CNN have been used here also. When you look on to this one, we'll check out with the accuracy. Here the accuracy is 0.6, loss is 0.676 and accuracy is 0.732, but here it is not so. The accuracy is very less because the complexity of the model increases. If the complexity increases, the input data should be increased. Then only it can read out the things very, it will make out the things, or it will find out the discriminating functions from the discriminating features from the images. So that is not possible here because I have used very less number of images here. So this model evaluate to be less accurate, both in my training and testing thing. And then I have also plotted my testing and training validation accuracy. So done with both these things. So if there is any doubt, we can discuss more.
Participants, any queries or clarifications required from your side?
Okay, ma'am, then we can wind it up.
Okay, ma'am. So thank you, Manali ma'am, for this wonderful and very clear hands-on session in creating a deep learning model for medical imaging. And the effort you took on for stepwise description of the model implementation was really useful. Hope the participants also had a very clarity on that. Thank you so much, ma'am.
Thank you, ma'am.
Participants, please fill your feedback. The link is posted in the chat box. And so I thank all the participants for showing the interest in participating in this pre-conference virtual workshop organized by MLRG group of SSN. And we request you to participate in the future events too and get gained, where we all share our research ideas and informations through us. And the e-certificates will be issued to you, provided your mail ID registered.
The CE certificate is purely based on your attendance. So those who have attended the sessions, e-certificates will be provided, because it will be dealing with, it will be dealt with this some other team, so we cannot do anything. So those who have given your feedback, attendance will be taken into consideration. So e-certificates is purely based on your attendance. It will not be given for all registered participants. Okay, then we will be conducting this IVA workshop, that is image, video and analysis. Vision, yes, this IVA workshop for every year. So I hope that if it is for the next year, we will have another set of expertise. Those will be discussing their new areas of research. So if you find any invitation or any such IVA workshop information, kindly please register and attend the workshop. Thank you.
Thank you all. Thank you again for participating in this event and made it success.
Thank you, ma'am.
Hope you all benefited too. Thank you all. Ma'am, let's close the session.
Yes, ma'am. I will tell Bala sir to do this.
Okay,
thank you, ma'am.
Thank you,
ma'am.
Mirnali ma'am, you can stop the recording.